वो देख लो इधर शुरू हुआ क्या होता है कोणा सोपानला झालं काम का इधर तरी कनेक्ट हो गया हो बघा बस ना म्हणजे आम्ही टाइम झाला निघणार सुरू कर सकतो ओके क्यों म्हणजे त्या कॅमेरा लावायचा का काय गरज नाही काय कोण लावायचं नाही याचं चालू आहे ना याचं कनेक्शनचं कसलं हे बघा याच्या पॉवरचं ना ओके हे आहे ना आणि याला आहे ना अमोल सर तुमचं वायफाय त्याला कनेक्ट केलं हॉटस्पॉट ते पण लावले समजा याचा प्रॉब्लेम झाला तर हॉटस्पॉट असेल नुसतं हॉटस्पॉट उभी राहणार कोणाचं तरी केलं तिघामध्ये कनेक्ट केलं हॉटस्पॉट हॉटस्पॉट कनेक्ट करतो प्रॉब्लेम चांगले जिओ वाला कुठे गेलं ते पण कनेक्ट करून ठेवा ना हे लाईनचा प्रॉब्लेम झाला जिओ चालेल ना लगेच ते कनेक्टच बघून घ्या हे शेअर करून ठेवा मग चालू राहू द्या ते चालू राहू द्या नाही काय होतं नाही लँडचं पाय तो एक मिनट मैं ये लगा देता हूँ।
इको आने आएगा ही वो क्या कंट्रोल कर सकता है?
नहीं नहीं, यहाँ का माइक मैं बंद कर दूंगा,
ओके।
तो सामने वाले को नहीं आएगा, बस यहाँ पे जो बैठा रहेगा
उसका आवाज आ रहा है।
हाँ, आ रहा है, मैम का भी आ रहा है, उनका भी आ रहा है।
आ रही
क्या
अरे डायरेक्ट
क्या
नीले का एक ही दाने जब वहाँ से
हाँ,
ये ईयरफोन चाहिए
हाँ, अभी ग्रीन पे ही है।
ये लगा के अगर चाहिए
दूसरा हात राहिला दूसरा हात असं नाही असं
अभी को नेट बंद हो गया क्या का प्रैक्टिकल लेगा
बस
बराबर है ना सर?
हाँ, सब बराबर है।
बराबर। धर्मेश भाई ये एक्स्ट्रा है,
एक्स्ट्रा? हाँ, एक एक्स्ट्रा है।
ये जगह किधर कुछ हुआ तो ये लगा देंगे ऐसा।
ये लगा दे।
ये नहीं प्रॉब्लम था ना,
ये थोड़ा प्रॉब्लम हो रहा है। चालू है अभी?
सब
हॉटस्पॉट पे चल रहा है, हॉटस्पॉट चल रहा है।
अभी कुछ मत देखो,
अभी हॉटस्पॉट चल रहा है।
चलो अभी मैं बैठ जाता हूँ। पहिला माझंच काय साहेब तुम्हाला इथं
जाएगा
बसायचं
तो ही इसका नेट जाएगा।
कोर्सर कमा गया हुआ है वो उस पे ये अब मेरा फोन फ्लाइट मोड आप इधर डेमो शुरू करो ना, अभी क्या हुआ?
हेलो, गुड मॉर्निंग ऑल ऑफ यू। एम आई ऑडिबल? नो।
शुरू करायचं ना, किती वाजता?
एक डेमो बघून घ्या.
कशाला? चालेल, डेमो किती होतं का? हां, जास्त पॅन गाडी गॅरंटी घेण्याचे काही तुमच्याकडे
स्ट्रीमिंग है
हाँ स्ट्रीमिंग चालू आहे.
हे इथं जेव्हा कनेक्ट होईल परत ते माईक ऑन राहील तेव्हा तिथं सगळी
एक दोन आवाज 4 लोक बाकी नाही.
चांदोबा चांदोबा भागला मोठा तुम्ही सरस्वती म्हणले ना तुम्ही स्वतः गाणार आहे. एक कनेक्ट करू सर इथं कसं आहे घोडा चाललाय गाडी हे बैलाच्या गाडी अभी स्ट्रीमिंग ऑन नहीं किया ना लाइव नहीं किया लाइव है हम लाइव सर नहीं हम लाइव डाल दिया लिंक पुन्हा कोणाला दिली
लिंक अभी शेयर नहीं है. जो मैम को सब्सक्राइब करके रखे हैं उनके पास ही गया.
अच्छा ऐसा है क्या मतलब दिख रहा है जिसको जिन्होंने सब्सक्राइब किया 800 लोगों ने सब्सक्राइब किया मुझे गुड मॉर्निंग स्टूडेंट्स
क्या क्या चल रहा है?
ये मेन कनेक्शन वो वाला है यहाँ से यहाँ से कुमार सर बोलेंगे और यहाँ से
मैडम बोलिए.
पूरा एक्सरसाइज करो
क्या करने वाले?
यहाँ से
चीज पहन के हो गई जीन्स पहन सकते हैं. आज का दूल्हा वही है इसलिए कोई टेंशन नहीं.
यहाँ से सब सर यूट्यूब से यहाँ से
ठीक. ये यूट्यूब लाइव डांस भी करेगा क्या? गुड. चलो तो मैं कनेक्ट होके देख लो. आवाज आ रही है.
हमने एक बार देखिए सर थोड़ा डेमो क्लियर
आ रहा है ना?
एकदम क्लियर. अभी तो लिंक शेयर कर दीजिए.
एयरटेल सर हमारा भी
नवरदेव कार्ड आज बघ यूट्यूबची लिंक अरे नाही दोन वाजता पहिले 15 मिनिटं माझं आणि मी काय म्हणतोय ते लिंक शेअर करून द्या ना आता. हा करून टाक झालं जॉईन होऊ दे ना. हा सगळ्यांना जॉईन म्हणजे आपला युट्यूब वर पण शेअर करून दे.
आप ये लगा लो म्यूट कर. मैं यहाँ से कनेक्ट कर लेता हूँ.
ये लगा लो.
हाँ
मतलब समझा नहीं
मतलब वो म्यूट कर दो ऐसा।
कंटिन्यूस म्यूट रहेगा।
हाँ
और फिर नहीं, मुझे फिर आवाज नहीं आएगी ना। मैं क्या बोल रहा हूँ, यूट्यूब लाइव चल रहा है।
हाँ चलो, हाँ वो चलेगा।
सुन दोस्त, आवाज नहीं आएगी। अगर इसका जब आवाज शुरू कर देगा तो फिर बाकी लोगों को डिस्टर्ब होगा।
हाँ वो भी है। ये मैं बोल रहा था इसका म्यूट कर दो ऐसा। ये लैपटॉप का म्यूट कर दो।
नहीं बोलना।
हाँ ये मतलब मीटिंग
हाँ, ये म्यूट है अभी।
हाँ ये म्यूट कर दो, मैं यहाँ से ऑन करता हूँ। आप जो भी बोलोगे सामने वाले को सुनाई देगा।
हाँ, लेकिन हमें नहीं सुनाई देगा ना, मुझे नहीं सुनाई देगा अगर मैं कुछ हो रहा है।
आप ईयरफोन लगा लो ना, इधर सुनाई देगा आपको।
तुल सर कुछ ऑन करो, चलता है क्या? देखो मैं यहाँ पे हूँ।
जैसे सर सुनते हुए बोल देंगे ना, ये ऑन क्यों कर देना।
हाँ बेल चलेगा।
हाँ
हेलो
हाँ चलेगा।
इधर भी रखो सर, नहीं रहेगा टाइट। एक बार देख लेंगे इसका आवाज आता है।
इसमें चेक कर रहा है
500 प्रवीण सर।
नहीं माइक
ईयरफोन निकालना पड़ता है इसके लिए।
माइक में तो आवाज आ रहा है। माइक में
लेकिन आ
आवाज
नहीं रहा है इसमें।
आवाज तो यूट्यूब लाइव।
नहीं ना, पर यूट्यूब ही नहीं है। हेडफोन की यहाँ आवाज आनी चाहिए ना, यहाँ से तो जा रही है।
हाँ यहाँ से जा रही है, माइक में जा रही है।
यहाँ नहीं आ रही।
आई मैम
नहीं, ये
वापस लगाओ।
नहीं दो। जब ये इससे ही कर लीजिए।
लेट आ रहा है इसके साइड।
अब मीट और तो बरोबर रहता है।
प्रवीण सर कैमरा ऑफ कीजिए।
रवि करता है।
ये लेवल देखो ना यहाँ
लेकिन तेरा ये बंद है ना, ये ऑन करना पड़ेगा ना। हाँ, पहले दिखा रहा है समझ में। हाँ, ये ऑन करना पड़ेगा।
बराबर अभी आएगा।
अच्छा नहीं आ रहा है। अच्छा नहीं आ रहा है, लेकिन कम आ रहा है। ये अच्छा नहीं आ रहा है।
अभी डायरेक्ट बैकग्राउंड हाँ डायरेक्ट लगा दो ये देखो। अभी तुम इसका माइक से
हाँ, ये अभी सही आ रहा है। तो एक तुझे ही काम करना पड़ेगा बस ये। ठीक है, मैं बैक करता हूँ। ओके, आता है तो व्यवस्थित है।
हाँ सर क्लैरिटी आहे.
तो आधी स्पीकर बंद करा.
ये ऑन है देख, इसका पासवर्ड भी बता देता हूँ। गलती से ये हो गया ना, तो इसका 1686 हाथ पे लिखिए।
हाँ 1686
आता नहीं, आता 60 सेकंड लगेंगे.
1686 ठीक है। गलती से अगर बंद हो गया तो
हाँ
नहीं ये तो है.
ठीक है। तो अभी बस मुझे लिंक फॉरवर्ड करो।
जुगाड़ पे ही है।
ज्वाइन नंबर काम किया गूगल मीट से लिंक फॉरवर्ड करके ज्वाइन कर दो।
हा गुगल मीट वाल्यांना करून टाका आणि युट्यूब वाल्यांना ज्यांनी
तुम्ही बसा
यहाँ पर मुझे लगता है यहाँ का जो है
नहीं यहाँ से ऑन है।
नहीं गूगल मीट का ये स्पीकर है
हाँ ये ऑन किया है।
बंद कर दिया.
नहीं ये जो साउंड है इसमें से बजेगा।
अच्छा कब चालू कर दें
बाद में बंद करो। किसका माइक चल रहा
हाँ ये चल रहा है, ये माइक चल
है? ये
रहा
बंद कर दो ना। म्यूट नहीं बोला, म्यूट करो उसको, म्यूट करो उसको। बस मैं जब बोलूंगा तभी उसको अभी ऑन करना।
अभी सब आवाज बंद हो गई.
कोई एडमिट होने आएगा ना तो एडमिट कर देना बस इधर
हाँ.
से। ठीक है। कोई नहीं बोले। नो इधर।
है। अबे कहाँ है? निकालना है तो अभी बैग निकाल ले फिर वो ना बैग निकालना अभी निकाल लेगा।
तभी निकल जाएगा। देखना
हाँ वही बोल रहा हूँ,
बाद में नहीं निकलेगा।
बाद में नहीं निकलेगा।
नहीं निकलेगा।
लोग सब जुगाड़ पे चल रहा है। ये तो है ना।
हाँ सर चल रहा है। हाँ, ये ठीक है।
तुमचं जेव्हा हे असेल ना दुसऱ्या नाही माझा मी काय केलंय प्लेन मोडला ठेवतो
अनिल सर को
एक अशी लिंब अशी
अब सही है ना?
हाँ सब चल रहा है।
सब मिल रहा है तो सब चलेगा।
अब इंटरनेट प्रॉब्लम था
ना बीसी लैब में लेते ना
ऍटलीस्ट त्याच्यावर सगळं सॉफ्टवेअर पाहिजे
जो पानी
हाँ ऑफ हुआ। भाई आया नहीं रिक्वेस्ट
आएगा अभी सबको भेजा
मग त्यांना पर्सनली
नहीं आया।
कोई नहीं
ठीक आहे ना तिथं हे करून टाका ओनली फॉर प्रेझेंटेशन मी मला नाव सांगा तेवढं जर आहे ना तुम्ही जे
ती जर डायरेक्ट पीसी बंद करतोय ना कर आवाज येतो मग त्या दिवशी आमच्या
एक मिनिट डॉक्टर रंजना सिंग
हा ऍडमिट
विजय प्रोफेसर विजय
येतो इनॉग्रेट
सिलेक्शन लोग भी आएंगे ना दिन का सेम किया है
नहीं सर दूसरे लोग भी हैं, बच्चे भी है उसमें
अपने प्रिंसिपल सर हो गया
यहाँ पे
आपल्या प्रिंसिपल सरांना
नहीं आया प्रिंसिपल सर
आपण एक्झॅक्ट 11 वाजता
कभी इसमें से देखते नहीं लोगों को presentation। 10 ही आज 10 ही लोग हाँ। हम्म।
हेलो सोपान
यस सर।
हाँ गुड मॉर्निंग सर
तो मागे मतलब हेलो आवाज येतो हो येतो आणि क्लॅरिटी आहे का आवाजाला येतोय का
किसके लिंक दिली हा डायरेक्ट इथे बसेल असं.
सत्यम पांडे नाहीये.
त्यापेक्षा त्याला तिथे बसवलं ना.
सर आप वहाँ से बोलो, आवाज आएगा यहाँ से, क्लियर आएगा, बस वो बंद रखना.
आणि हे बंद आहे.
सत्यम पांडे
हे डिना अवतार देतो आम्ही.
सर कर ठीक.
दोघे एकच आहे ना.
हाँ एक ही आईडी है.
मग एवढंच पण ऍड करायचं.
हे मला माहिती आहे.
तुझं जेवढं list आहे. आणि तिथे त्या ग्रुपमध्ये
जनरल इन्स्ट्रक्शन असतात त्यांना नाही पोहोचत. इसको हिलाते रहने से.
नहीं वो खोल दो 1686.
हाँ बदलना पडेगा. हो झाले. अरे कारण ते पण एक्झॅक्ट 2 मिनिटं सरांना प्रिन्सिपल सर झाले करून सरांना जॉईन व्हायला पडतं आणि आपण चालू करून घेऊ. प्रिन्सिपल सरांना फोन करून सांगू सर जॉईन व्हा.
तुमचा आवाज ऐकतो कोई मैसेज किया, कमेंट किया गाना सर.
ओके सर.
कमेंट
आप
किया.
मुझे डिनाय करना जो भी करना है.
ज्यांना डिनाय करायचंय ना तर कोणी आहे ऍडमिट करणार.
मी सांगतो ना नाव मला फक्त सांग. मैं जब बोलूंगा अनम्यूट करना फिर ये
वो बंद हो गया.
बोट कापून देऊ.
उमेश
अभी ऑल कर पहले.
उमेश
हाँ करो करो add करो.
आणि संतोष
संतोष क्या
इम्पत
हाँ संतोष ऊपर को करना.
वो चला गया.
आ जाएगा वो.
चलो चलो
सब बड़े लोग हैं, चले गए ऐसे बताना, चला गया ऐसे बताओ.
ये करायचं होतं ना. साइड चेंज करून बघू.
काहीच नसतो सर जॉईन झाले. तुम्ही मला फक्त विचारा कारण मी हे केलं. माहिती करायची गरज नाही सर.
ते पण बोललं तरी भाग
काही लोक बोलतील पण
इथे तर हे फक्त इनॉग्रेशन आहे. ज्यांचं टॉकेशन नंतर
इनोव्हेशन
आपले बाकीचे सगळे ऑन युट्यूबवर आहे. हा लिंक देऊन टाकतो. यूट्यूब अभी हो गया ना?
हाँ वो तो चालू है.
तू पटकन देऊन टाक. तू आतापर्यंत देऊन टाकाल बघ.
काय हरकत नाही. युट्यूब वाल्यांना काय नाही. हाँ यूट्यूब पे आएगी क्योंकि वो माइक चालू रखा है यूट्यूब का. बंद कर दो. हाँ वो यूट्यूब पे चलेगा.
यूट्यूब पे बंद कर.
2 मिनट के लिए बाद में चलेगा.
जो मीटिंग में उनको
जब
नहीं
लाइव
सुनाई
चालू करा
देगा.
जब सर बोलना शुरू करें.
दीप पटेल.
नाही.
दीप पटेल.
नाही.
नाही करू. एक लक्षात ठेवायचं. हे जनता गिगाबरोबर.
त्यांचे कामात बसले. त्याची नॅशनल स्टार्ट करू ना.
स्टार्ट करून घ्या.
हा स्टार्ट करायचं.
सुरुवातीला सर
तुमचं कुठे पण स्पीच होईल.
माइक ऑन करून दे. कर दे. प्रिन्सिपल सर बोल. गूगल का ऑन करना है. माइक
झिरो करू. मला कसं
हाँ तो सर बोलेंगे ना उधर से. हाँ तो सुनाई देगा. सर सुनाई देगा सबको. सुनाई
बस यूट्यूब पे तुम्हें
वालों को नहीं दिखा सुनाई देगा ना.
चालू कर देंगे ना जब बताता
नहीं
हूँ.
यूट्यूब पे सुनाई देगा.
ऐसा है क्या?
हाँ
मैं बताता हूँ तब चालू करना उसका.
तो यूट्यूब का भी बंद कर दो.
हाँ बंद कर देना.
रोजी खान
नहीं.
दोबारा आया.
करो उसको. ठीक है टाकलं. झालं सरांना बोललो. हाँ ठीक आहे.
चेक करना है यूट्यूब पे. ज्वाइन मीटिंग.
आणि ग्रुपमध्ये मेसेज टाक. we have started.
यूट्यूब पे वॉइस चालू करना होगा तो बोलना. हाँ यहाँ पे भी आ रहा है. यूट्यूब पे आ रहा है.
यूट्यूब चालू कर.
आता है. संतोषी पर
A very good afternoon, good morning to one and all. I, Swidar D'Souza, Assistant Professor in Botany, take this opportunity to welcome everyone gathered here virtually for this National E-Conference on Recent Advances and Opportunities in Botany, NCREO 2021, organized by Department of Botany, SSR College of Arts, Commerce and Science, Silvassa. Every good work should begin with the blessings of the Almighty. So before we start, let us invoke Goddess Saraswati and seek her blessings.
या श्वेत पद्मासना या ब्रह्माच्युत शंकर प्रभृतिभिः देवैः सदा वन्दिता सा मातु सरस्वती भगवती निःशेष जाड्यापहा।
Now, I would like to introduce to you our principal, Dr. Rajiv Singh. Dr. Rajiv Singh, sir, is specialized in the field of entrepreneurship development. Sir holds a PhD and has published many research papers nationally and internationally. Sir has published 12 books internationally. He has served our country as a Navy officer and later joined in the field of education. He has won the Best Principal Award in 2019 and was the winner of International Award of Excellence in Research in the year 2020. I invite Sir to formally address this virtual gathering. Over to you, sir.
Thank you, ma'am. Thank you. A very good morning to all of you. Am I audible, ma'am?
Yes, sir.
Very good morning to all of you. The National E-Conference on Recent Advances and Opportunities in Botany is going to be organized by the Faculty of Science, the Department of Botany of SSR College. The SSR College, governed by SSR Memorial Trust. The trust in a class of its own. The most renowned visionary and our source of inspiration, Honorable late Shri Mohan S. Rilkar, who recently passed away. He was our chairman. So the whole this knowledge sharing activity dedicated to him. The SSR Memorial Trust is always been dedicated for the quality education, providing sound knowledge with as far as concern, as for providing the good quality infrastructure and source of knowledge. We have more than 100 faculty in the whole SSR Memorial Trust. The SSR Memorial Trust govern 4 colleges under their umbrella. The SSR College of Arts, Commerce, Science, SSR College of Pharmacy, SSR College of Education, and SSR Institute of Management Research. We have Arts, Commerce, Science, Pharmacy, Management, and Education specialization in the campus. As far as SSR College of Arts, Commerce, and Science is concerned, recently we accredited by the NET with B+ grade, and we continuously working for the quality. In the same manner, this whole conference is going to be organized. I would like to welcome on behalf of SSR Memorial Trust and SSR College of Arts, Commerce, and Science, the renowned speaker of the program, Professor Vijay Mandurkar, the Registrar, Dr. Homi Baba, State University, Mumbai, Dr. Sanjay Jatta, the Joint Director of Higher Education, Panvel Konkar Division, Professor Avinash B. Ade, Head Department of Botany, SSR, Savitribai Phule Pune University, Professor Arvind Dhabe, the Head Department of Botany, Dr. Baba Saheb Ambedkar, Marathwada University, Aurangabad. These are the today's speakers. And tomorrow, Professor Umesh Kakle, Department of Botany, the Institute of Science, Mumbai. Professor Sangeeta Godbole, Head Department of Botany, Jai Hind College, Mumbai. Professor Dr. Sunil Sangle, Department of Botany, Rajaram College, Shivaji University, Kolhapur, and Professor Dr. Ranjana Singh from Government Model Degree College, Bulandshahr, going to be joined tomorrow. This is one of the key factor of this knowledge exercise. Apart from that, many participants are going to join this program for presenting their papers and posters in different sessions. Dear participant, I would like to congratulate that this conference is going to be next milestone for your professional life. So you may get the knowledge and share your experiences also with the through the feedback, as well as if you require any kind of query. So there is certain links may be provided by the organizers or the team, and I hope so. This conference going to be next milestone, next benchmark for the success of SSR. All the best to all of you. Thank you so much. God bless you. Over to you, ma'am.
Thank you so much, sir. Now I would like to call upon the organizing secretary for today's national e-conference, Dr. Sopan Parat sir, to briefly introduce to you the theme for the national conference. Over to you, sir.
Thank you, ma'am. Good morning, all. I, Dr. Sopan, Organizing Secretary, welcome you all to e-conference. It is our immense pleasure to see you all across the screen. I will be walking you through what the conference is about, how useful it is for you, and what does the future holds for all us in respect of the theme of conference. The Department of Botany, SSR College, Arts, Commerce and Science, Silvassa has organized conference on recent advances and opportunity in botany. This is the first conference in the history of our institute which is completely conducted on digital platform in line with social distancing norms due to COVID-19 pandemic. The speakers and delegates who have joined from all over the country today to share their knowledge are indeed going to enhance our intellectual tourism. We have the speakers and delegates who have joined for all the country, okay? We are enough fortunate to have highly profound delegates with us, namely Professor Dr. Vijay Mendulkar, Dr. Sanjay Jagtap sir, Professor and Dr. Avinash Ade, Professor Dr. Arvind Hade sir, Dr. Sangeeta Godbole, Dr. Umesh Kakade sir, Dr. Sunil Sangle, Dr. Ranjana Singh. Today's conference has about 170 plus participants from across India, making this conference a truly national conference in spirit. We are proud to announce that conference is being led and guided by renowned personality in the field. Information about this conference. This e-conference is scheduled for 2 days, having 2 technical sessions for each day. On the day 1, we have total 4 eminent speakers in technical session 1, and in technical session 2, total 9 research paper and poster presenters. On day 2, again, we have 4 eminent speakers, technical session 3rd, and in technical session 4, total 10 research papers, poster presenters. This is the brief introduction to e-conference. I would like to thank our SSR moderators and principal sir for giving us this golden opportunity. Thank you once again. Once again, I welcome all the eminent speakers and guests from all over the country and from different walks of academic life who have joined to share their knowledge and vast experience to the entire community. I am also very much thankful to all my respected beloved teachers and eminent speakers who gave me a confirmation to join on a single call. And it is their blessings on me by sparing their valuable time for me, all of us. Thank you once again. I would like to conclude my speech by thanking all the delegates who have participated and are going to enhance the level of knowledge by all the activities and discussions through the digital platform. I am thankful to all participants, research scholars, BSc, MSc students, my friends, my colleagues, and all the technical team supporting us. Thank you once again. I wish everyone a successful, safe, and fruitful conference. Thank you all and happy learning. Over to you, ma'am. Thank you.
Thank you, sir. Now, let me introduce to you the keynote speaker for today's program. Our keynote speaker for the conference is Professor Vijay D. Nidulkar, Registrar Dr. Homi Baba, State University. Professor Vijay D. Nidulkar is the Registrar of Dr. Homi Baba State University, as well as the head of the Department of Botany at the Institute of Science, Mumbai. Sir holds a PhD degree and is a recognized guide for the subject of botany and biotechnology. Sir is specialized in the area of cytology and genetics. Under his guidance, 6 students have completed their PhD and currently 7 are working. Sir has also guided 1 postdoctoral student. Sir has published 68 research papers in various national and international journals. He has attended as well as presented his work at more than 68 national and international conferences and has organized 4 conferences. Sir has more than 28 years of teaching as well as research experience. He is also a reviewer for the journals Material Science and Engineering C, International Journal of Pharma and Bioscience, Journal of Applied Life Science, Journal of Material Science and Nanotechnology, and International Journal of Advances in Plant Science, and is also an active member of various national scientific committees. Having such an excellent person with us is an honor. I would like to ask Sir to commence the conference and begin with his lecture. Over to you, sir.
Thank you, madam. First of all, I think this is a very good opportunity to all of us to come together on one dais, and the credit goes to the organizers. Especially, I would like to appreciate the initiative taken by Principal of SSR College, Dr. Raju Singh, sir. Sir, your interest, I could make out your interest in starting or initiating such activities to promote the idea of research, in spite of having such a critical condition, pandemic situation. We are not going away from our aims and objectives, and we are strictly adhered to whatever way and whatever way we wanted to contribute. We remain to do it either by organizing conferences, discussions, or workshop of this kind online, and that is creditable, sir. This is not only creditable to your college, but I think it is for all of us. Secondly, I would like to appreciate the initiation taken by Dr. Sopan Kharat. Of course, he has invited really a good team of speakers for your college as far as this subject is concerned. And as I know, I could see here Sagarika madam and my best friend, Professor Dhabe sir, then Ade sir is also there, Takade sir is also there. So I think this conference will be really a fantastic one. At least we'll know each other, and we'll share some views and some ideas so that it will be benefited to all the members who joined this conference. Now, the time when I was approached by Dr. Kharat, I was asked to deliver one speech and relevant to the concept of theme that you have designed for today's meeting. And I thought of to just generalize the thing, but with useful information as far as techniques, methods, and other things are concerned, rather than focusing what someone has done in what aspect, because that becomes a matter of individual credibility. And the conference, when we talk about, it is always good to impart the information which will be useful for others, newcomers, and other teachers or students also. So keeping this point in mind, I just requested him to permit me to talk on this particular issue, which will have some information about the scenario of research activities. I'm talking about the scenario, research activities in past and present, or in other word, we can say what are the changes, not in terms of techniques and methods only, but the changes in terms of views, tendencies, strategies, policies, and concepts. As far as our research in our subject is concerned. So that point of view, I'll be happy to share with you a few points. And if you permit me, I can start that.
Yes, sir.
Okay, right.
You can start.
Yeah, right. I'm even happy to see that some of the guests from other state also joined. Dr. Ranjana is there, who is very much known to the institute and to all of us. So I'm very happy.
Thank you, sir. Good morning, sir.
Good morning, Ranjana. Good morning.
I'm also happy to see you, sir, after long.
Right. Sangre sir is also there. So anyway, we shall start our this one. Today I would like to focus on the investigation or approach of, rather I can say approach of investigation in botanical research, past and present, methods and techniques. Now in this, I would like to just summarize that whatever we say research today, tomorrow it is past. So what is the durability of saying research? In other words, it is very limited. For a limited span of time, the word research, recent can be considered, because what we consider is recent today, tomorrow it no more is recent, and again it replaced by something else. And this kind of transitions or modification have taken place in almost all the subjects. Not only botany, I'm not referring this our subject only, and almost all the key speakers are research oriented. They are having very good, and I would say that excellent background of research, and they're well versed in all the methods, whatever they have applied for their work. So they will be certainly agree that the methods and techniques that we used at our presence during our time, those methods were recent at that point of moment for us, but then equally same point during same period, all those methods were past methods for the one who started working before us, some 30 years before us. For them, all those were past. So if we go on just cataloging the things and the just referring the approach and the transition and modification in the methods for similar aspect, you take any parameter for that matter. Suppose we are performing any karyotypic analysis, the way it was performed earlier was different. The way it was performed in between, it was different. The way it is performed now, it is different. The parameter is same. We are studying karomorphology. Those time also the people used to study this karomorphology. Now also we are doing same thing, but then the approach is different. So when we talk about the recent advances and present and past methods, then what are those modifications, what way it has gone tremendously and what way it has changed. That really becomes the matter of thinking, and that's why it is we have to summarize the things, that the techniques, what way they have added or what values they have been added, these techniques, modern techniques and methods, whatever we are adapting now, what values been added in the research on working on same parameters. And if there is addition of some value, there is addition of some importance, there is addition of the details, whether that details are contributing constructively for formulating our aspects or designing our research in today's area or not. That is the issue, and that way it will be benefiting. So that I think I have concentrated on that issue specifically. Now, especially, of course, you will find here the reflection of botanical methods, because being a person of that discipline, I have highlighted, but this is applicable to other subjects also. Especially, see, when we talk about very, very old days, the herbal drugs, serendipitous discoveries I have written, actually, there was no serendipity, because that is serendipity for us only with respect to innovation at our level. But it was invented earlier by traditionally some Vaidus. So herbal drugs are not new things for us, right? That time also, the people used to study those things, used to collect the samples, they were knowing applications, they were knowing the part of the plants, how to use, when to use, what for it is used. So for us it was surprising, for them it was not surprising. But because we exposed those things at that point of moment, we started realizing that it is surprising for us. So we are representing today's era, or today's scenario. So we focus ourselves only. And when we focus ourselves for any research activities, then I have to say that it is really a serendipity for us also, surprising things for us also. So later, some innovation started, then new eras or new aspects surfaced out, cell studies or discoveries on vitamins, investigations on vaccines. Then 1940s, there was a talk about antibiotics at preliminary stage. Then research and development activities were boosted at that time, and many activities were conducted against many diseases. Then when we talk about 1970, there was a little bit, to some extent, not 100%, but that was a period when the biotechnology started coming up with few aspects, and the researchers also started applying those techniques in their respective fields. And none of the fields that is left behind where you we can say that there is no biotechnology, use of biotechnology. Of course, it depends. But if you want to apply that concept, then almost in all aspects, these biotechnological concepts can be utilized, used, and useful results can be generated. But then I would say that it is not always necessary to go for advanced things, because many important investigations been done based on simplicity, and that is sort of good guideline for us also, because always we should not go for sophistication and sophisticated steps, methods, techniques, etc., etc. But then this is the fact that the research activities nowadays will tremendously changed by incorporation of different methods and techniques. Then when we talk about 1960s, that was the period when we born, then there was slightly advanced knowledge, and it was generated by group of people having good information about all round parameters of the materials. Let it be a biological materials, let it be we can say other materials also. But then there was the extension of sort of activities. Then further, the discovery of DNA. Now, discovery of DNA, what we say, it is okay, fine, but it's not related to only to the extent of its structural elucidation or its importance in genetics. It is related to the existence of some of the genes which are causal factor for diseases or genetical disorders, for the study of genetical disorders. And see, there is no question of poor and rich. There is no question of high society, low society. There is no question of officer and mazdoor, because the system is same for us. We all are we all are surviving under similar condition, under similar structure, and the system is governed metabolically, genetically by DNA in all the cases. So we see that point of view, wide range of thinking. If we apply here, then we'll realize that this particular component, genetic material, is the mastermind material, and it has many things to do, or it has the roots of many such things which need to be focused and needed to be focused, and it was focused already in past also. But then nowadays, if you see the techniques being used for focusing such disorders, genetic disorders, targeting the DNA, then you will realize that it is tremendously been changed, and new techniques are developed. Now we have gone up to the level of arresting activities of messenger RNA when it was not under consideration, or it was difficult to believe. It was okay up to the level of DNA, but we have gone beyond that now. Then in 1980s, commercialization of drug discovery, combinational chemistry. Why it has come? Because of the innovation and techniques. Now, what techniques lead this commercialization of drug discovery? Now they are not in raw form. They are well isolated. They are extracted. They are optimized. They are purified, and their reactivity, or you can say their action, also perfectly been clinically tested. Now, once it has been done, then naturally the multinational companies or pharmaceutical companies or any other establishments who wanted to make a money out of it, they started coming on front page, and then started pouring a lot of amount, taking manpower, developing skills, and appointing some skilled persons and getting the product in their own establishment and marketing the things. Then the robotics also have come, and now this robotics, of course, it is not new thing. Almost all we know that they are now taking the help of robots for operation also. So this is how the changes, we can cite the changes in this area. Now, general principle of any research, and if we cite the example of especially herbal research, which is the attracting and focusing point for all of us, appealing point for all of us. Now everybody will believe that the generalized plan, it is normally anybody when select the material, or will when he consider the material, then we see its taxonomical details, we see its fingerprinting, chemical fingerprinting also we study. We also study toxicology. Then there is approach, ethnobotanical, ethnomedicinal approach. Then we go for the information management, where it has been used, whether it has been catalogued, whether it has been registered, whether it has been in use in other sector of the society, somewhere in other geographical region or what. So all this information is needed to be collected. This is what the sort of flow sheet we can say, or the pattern where when we begin our things. Then the plant extract is prepared, extraction, toxicology, we test toxicology, we perform bioassay, then pure compound is prepared, its preclinical testing is done, then clinical testing is also performed, and lastly we go for the marketing. So marketing not our business. This is the commercial issue. So this is done by somebody else. We provide the basic information on these aspects are material. Further things, commercial things and other things, financial thing. This one is the matter of other organizations. Now, when we start working on any drug discovery, then the flow of methodology, you can say, or the techniques. Normally, it goes like earlier it was blind screening. Even now, also we just dig out particular wild plant or any other plant, and we feel that maybe this may be having some important target compound. We are not sure whether 100% that target compound it is there or not. We are not sure. So we are investigating. This is investigation. We are not knowing that, and we are just investigating the things whether it is there or not. Certainly, sometime we will get it, sometime we will not get it. But then that was a blind screening, or this is a method of blind screening that we adopt. Even this happens in genetical experimentation also, where the blind mutagenesis is performed without knowing which character responsible for which gene. We randomly we treat the things with mutagenic chemical. We induce the mutations, and when the phenotypic appearance comes into the existence, then we realize that, oh, so this is the particular character undergone change. That means what? The gene controlling that particular character probably might have gone mutated, or it is mutated. So the conclusions are later. Earlier we are not knowing anything, so blindly we are doing. Now the situation is not like that. It is now directed mutagenesis system is there. Site directed mutagenic experiments are done. Target is decided first of all in the plant mixture, and its screening is done now. The blind screening is it has gone now. Now for tissue culture also same thing was there. We used to propagate the plant. Let it be whatever plant. If the propagation is there under in vitro condition, okay, fine, it's good. One thesis is over. That means that was the chain of pattern of working in that field. Now nobody goes to propagate the plant in tissue culture plant. We have gone beyond that. We never go for the propagation of the plant. That technique has gone. And now if somebody is doing, it is not appreciated, and it is certainly the things will be coming that no, it has gone very old thing. Now you could have gone for some new techniques. Now further then it's bio transformation process is there. Then optimization of secondary metabolite under in vitro conditions are there. So many techniques now people are doing when we talk about the tissue culture experiments. Now those days of propagating and multiplication of the plantlets has gone. Of course, gone doesn't mean do not please mistake me that we should not do it. It is sometimes it is a need of ours, and we have to do it because sometimes it is warranted, it is needed for rare plant and all that. So that is the only way that we have to go by tissue culture plant to maintain them. Now, my point is that it was so blindly, it was we were working earlier. Marketing also, the method of marketing and the commercializing the product, that was also different during those days. You yourself can visualize, or yourself can be judge. You can judge best way. What way there is a change, a tremendous change in commercializing the biological products, especially biological product I'm talking about. Now at middle of 20th century, the screening has been changed. We have gone for not only pure extraction, then the concentrated screening we have done. System approach also been changed. Then target molecule been identified. So lead candidate is identified. Then it's present existing. We started not believing in that. So what we started working on, that to enhance its quantity. I'm not talking quality. The quantitative changes under in vitro condition already people started. And that has become a new approach. It was at that point of course, now it is different, but then this way it has changed. Then there was a preliminary development, then complete development of the protocol, and then there was a marketing. Now, many drugs which are used for chemotherapy treatments from plant, this one, they are very, very pure, and they are concentrated, and they have got a lot of values. But of course, this method of purification also has been developed, and they have been rectified in such an extent that it is 100% pure, and it can be clinically administered without any dilemma or without any doubt. So during olden days, all these methods were there. Extraction, of course, very common. Infusions, common. Decoctions, common thing. Tincturing, common thing. Ash also is known to us. Poultices, known to us. Snuffs, of course, snuff. Nowadays, it is not that much in a practice. But medical point of view, taking snuffs also, that during those days had some importance. It was not proper, it was not advisable to go for that. But old people used to do it. They used to get some relief. It is a stimulant or stimulation to the nervous system or whatever it may be. But to that extent, it was considered that it is important. Nowadays, nobody will believe that. Now there are other techniques where this particular concept has gone. Then guels also, we used to prepare the pastes and other things. So if we go for these different patterns, then we are not taking blindly. Any concept we are not taking blindly in a hand. We are not nourishing that way as if that we have touched and we have done. No, never. We never do that. So what we do, we see the background. We see the supportive work, whether been done or not. So then the question of literature review comes into the picture. Then its evaluation comes into the picture, to what extent they have done and what extent we have to go ahead. Now suppose a particular compound, andrographylide, is there in particular plant. People have purified, extracted, fine, they have stopped there. So if I wanted to go on that because of the attraction of that particular component in that plant, I will not go for that as a end point. I will extract and I will further go to see its activities. Then next person will, in next era, he will see that how that andrographylide can be enhanced by using different kind of elicitors. Now, this way it is step by step. The things are goes, and then the innovation or the pattern of innovation changes. This I wanted to just highlight here. So then toxicology has also been studied. Biomass protection is also that aspect is also very, very important. Then one important fact is that we are not disturbing the total biomass of the plant. A piece of the plant is enough for us. Now, earlier that was not the case. If the plants are located at particular place, it has to be uprooted, it has to be processed, it has to be, you have to process it by cutting its leaves, branch or stem or something like that. So that way it was a danger, sort of a danger, and against maintenance of biodiversity. That has also been worked out here, and now that is almost, it is stopped now. So herbal correction also is stopped nowadays by all the universities. So this is a very important thing. Because why? Because the new techniques are generated, and that same thing can be done by those techniques. So the clinical studies are done. Then the stability of components can be determined. This stability and maintaining the particular compound for a long time, this was not the object during those periods. And even if it was there, then it was doubtful, and it used to get denatured. Now you have got a lot of techniques to maintain it for a long time in original form and sustain its activities and retain its characteristics so that it can be used anytime with any concentration for the preparation of drugs or any other things. So this, as far as when we talk about the botany, then this kind of transition has taken place not only in the field where I am talking about or we are having expertise, but this is equally been adopted in morphological concepts, cytological concept, histochemical methods. For example, I would like to refer here that during our days, then the people or the researchers who did this kind of histochemical work, they are working in USA as a professor of histochemistry. But the work based on that or whatever the PhD concepts they have worked out and they have got their opportunity, it was based on simple techniques. I witnessed those things because if they wanted to report that in particular stage of plant flowering, if the content of DNA in the plant is very, it increases, it is varying, then how to indicate it? The way now we are indicating, that time that technique was not there, so they were indicating by histochemical analysis. So there are different type of specific stains. They will stain it. Microtomy was the best for them. They will take a stain. They will stain the slide, and on the basis of the intensity of coloration, on the basis of the density of coloration, they used to say that, see, the activities of this particular DNA molecule at this particular part of the cell is more. This area is showing less. So similar way for RNA, similar way for any other cellular components, they used to do it because that was the only way and that was the standard way. That was standard means it was appreciable way. Those, because that technique was everywhere during that period. But we now rarely go for microtomy because we have got so many other standard techniques. So my point is that the way which was earlier adopted by people or known people, now it has gone tremendously changed. Same is the thing is for anatomical sectioning also. Simple compound to high resolution microscope we have gone now. Laboratory morphology, experimental morphology, physiology. Now chemical fingerprinting is very much, means every one of us, let it be having that specialization or not, it is in matter. But the students are very much aware about this kind of terms like what is fingerprinting, what is chemical fingerprinting, what is chemical analysis, etc. What is phytochemicals. They are very much known. In biochemistry, different type of assays have come. Now, earlier testing has gone, methods been modified. In this subject also, there is a tremendous change. Same is the thing with the biophysics, preparation of instruments of biological related things with the physiology, with the help of physics discipline. Then in ecology, just surveying the things or studying the field. Now those days has gone now. They have become primary nowadays. So then there is a remote sensing has come. Then some other techniques also has come. There are the instruments to find out the density of the forest. There are the instruments to find out the release of gases and intake of gases. So those were not at that time, and the people started using all these instruments with the advancement, and separate institutions has been constituted or established by the government and private organization also. So there is a tremendous change now. In classification also, everyone of us know that a traditional method of classification has gone now, but I don't say it has gone now because that becomes a base for classification, whatever methods earlier we used to use. But in addition to that, some recent techniques also have come at molecular level, genetic level, then chemical level. All those things are, we are nowadays focusing. So as a matter of studies in advancement in this particular area. Same is the case with the prokaryotic studies and bacteriology, in even fungal biotechnology. Fungal biotechnology was not that much popular earlier. Now it is tremendously popular among students. and fungal enzymes. These compounds have got lot of importance, and many people are studying, especially by extracting these enzymes for many activities. So similarly, this, as I said, that in morphological aspects, grouping, handling of material for sectioning, and using this what is the dissenting microscope. Earlier, everywhere, for every ship you have to keep it. Nowadays, very rarely we see. And even in my department, I could see very few such dissenting microscopes which were purchased earlier long back, and they are maintained in good condition. Otherwise, it has gone, and nobody is giving. And because they are supplying, but it is with advancement. There must be some advanced technique. But if you see the old microscope, only simple plate, this objective and mirror, that was enough, and that was the base also for the evaluation of certain things. Now those things are not there. So optical evaluation has been have taken place. Microscope, there is a tremendous advancement. Binocular and apochromatic lenses, in development of lenses which are used for the investigation or in investigating apparatus, they have changed, their configuration been changed, their image magnification also has been changed, and we are taking the use of all those things. Now this kind of microscopy, sorry, not this is not microscope, this microtome. We used to have this microtome. Every department, every institutions who are performing research activities, it is, you will see this in their botany department, this microtome is the best way of taking the sections. Sections means of what? Section of any kind. Very hard sectioning, very soft sectioning, very, even the stone sectioning, or soft microtomy, that is called as sludge microtomy. Then all kind of sections can be taken and evaluated. Now the things are not there. If it is there, it is unused condition, is placed in unused condition. Probably some people for some specific things might be using, but those days are not, nowadays it is been rarely used. So this is the details of these microtomes, where we can have the sections of different thickness and the area where the applications can be studied. So now the advanced level microscopy is in existence. Everybody, whosoever comes to market or to convince that, no, say they are they are convincing, say, sir, this is microscopic having this kind of magnification, and this has been advanced equipment. So you go for this, you go for that, and we also, as a teacher, our knowledge is also, of course, basically teachers are concentrating on teaching, but research point of view also we are alert, and that alertness has some limitation. That I would like to emphasize here, because of some liabilities, or we are more focused on the other things also, teaching related only. Then the research related are not that much. We cannot behave like a pure scientist of a scientific institution, but it doesn't mean that the teachers are totally ignored about all these things. There are teachers, and the teachers having the knowledge to the extent they can say that really this is the advanced component. Spectrometer was, earlier it was of that level, and today it is of this level. That means what is the difference between that period and this period? That particular kind of difference, he is able to evaluate, and he is able to convince, and he is able to get that instrument. In addition, equipped with this kind of advancement in that particular equipment, So that at least that knowledge everybody is having. Every teacher who is involved in the research, they do have this knowledge. So my issue is that nowadays the faculty of different institutions or universities or colleges, they are also wise enough and they are also aware about the advancements in the research and the techniques and methods. And because of that only all these things are going on. Otherwise, it was very difficult to expose the student to the recent techniques and recent aspects. After all, when we study the aspects, the students are getting more value when he is having some information at advanced level. Primitive information is for knowledge and it is not for the execution. Primitively, nobody execute that information. Execution of the information, when it is there, then always advancement comes into the rescue. And that advanced information, as well as I'm talking about exclusively research related activities. When our student goes to other institution, then this matter comes into the picture. So the advancement, when they see that the students is having this kind of information, which is fine, it is justifying today's need, certainly his candidature is considered or he is admired. Otherwise, primitive is primitive only, and that remains to the extent of the knowledge only. So this is the issue. Now, when we talk about the metamorphosis in the optical analysis, I can say, then so many things are there, different types of microscopes are there, whatever are listed, they are also old one. I don't say that they are very, very advancement, because nowadays we are having transmission electron microscope, scanning electron microscope, so many things are there, X-ray diffraction studies are there. So people are going for that, and that is a need of ours, because the characterization of anything, characterization of any material, let it be biological things or any other things, need some touch of application of advanced equipment and utility of advanced instruments, so that the details can be referred very easily. Otherwise, it is just theoretical and is not that much appealing. Now, chromosome related things, if I say, there is no point in saying that no, I don't have interest in the chromosome, because you may not be having interest in chromosome, but your body is having chromosome. That cannot be ignored, and that is a fact. So when these studies on chromosomes started, it was simply on the staining method. You use acetosine or acetocarmine, especially for mitosis. We used to study that. Now, normally it is just studied or stained only to know the or to identify the chromosome, but the basic details are evaluated by bending pattern, by fluorescence studies, by labeling the DNA, by isotopic use of isotopic material. So many things are there now, and now you have started getting 3D photographs of chromosome, which were very rare and which were not exist during those periods. When we were working, we were very eager to see what we will get that picture, very good picture. Even the photographs of good somatic metaphase is there. That was sort of it was very good day for us to get that kind of things in any literature or any other thing. But then it was very difficult to get this kind of things during those days. Now it is very common. Any student at student levels also, 12th standard students also will be having a very good photographs, original photographs of time analysis and other things. So we have changed to that much extent, and therefore the investigations on location of genes on this chromosome and structuring of heterochromatic region, structuring of that DNA strand, this kind of things has come up with very fast, and that helps us to localize a specific sequence for specific gene which is responsible for the specific disorders or specific reactivity in terms of production of the enzymes or proteins. So many changes are there. This is a similar kind of things. I wanted to show you how the bendings and coloration and fluorescence studies been done. This Q bending, G bending, R bending, C bending. So different kind of bendings for different kind of observations, different kind to study different kind of effects, and also been to apply those things and to identify different kind of locations, a specific location or specific activities can be studied. So this was earlier setup of our work. Now this nobody will believe. Select the bud tips or this root tips of plant, preserve it, and prepare the slide. So badly stained slide. This kind of staining nobody appreciates now. Nobody appreciates now because it is something. It appears to be very, very primitive type of things and processing. So nobody appreciates now. Now people have one very clear views, clear lenses, clear photos, and purified staining reagents and agents. Those all things we have to apply nowadays. Then only our work is appreciated. Now, talking about different techniques in the identification of desired DNA strand, identification of desired mRNA strand, that mRNA is assay what we are doing, or that is also called as RNA's protection assay, where the things can be done what we are doing with the DNA at nucleus during the transcription. The things also can be done in the cytoplasm when there is a translation of the RNA factor into the protein. So you do not allow the you do not permit that mRNA stands to translate into the protein. So protein will not be synthesized. Even by mistake the gene has been transcribed, it will not be allowed to translate. So that techniques also has comes into the picture. Southern blotting is there, northern blotting is there. So many things are there. RAPD, RAPLP, ALP. Now these things are advanced. Why I am taking? They are not very much common. They may be strange to some of us, but these are the techniques which are people are in this particular area. They say that it is advanced technique, or this is a sort of advancement with which we, whenever possible, we have to start with, and we have to come up with the advanced conclusion. So we will be justifying the scenario of today's research and today's activities, research activities. Now, as far as assays are concerned, macro assays, different kind of macro assays we are doing, antibody, antigen reactions we are studying. Now, COVID is the best example for us, where normally the, as we know that there was even the problem with the detection, with the formulation of drugs against COVID. Now I think so fast and so aggressively, the scientists in different laboratories, they started working, and they have developed some drugs, not completely curing the COVID, but they have produced the vaccine. That is different thing. Now, the testing part which we are doing against COVID, as far as my information goes, that is real-time PCR we are using. And why it is being preferred? Because you can produce the results within 3 hours. So RNA has been this one is samples are taken, RNAs are extracted, they are hybridized, they reverse transcription, they are processed, they are subjected to reverse transcriptase, so the reverse transcription, then DNA is prepared, and then the by hybridization process, if that particular COVID genome is there, it will have some alignment, and then there is a confirmation. Of course, PCR analysis also many cycles they are doing, more than 30 to 30 or 40 cycles. And then if it is matching, then they conclude that yes, there is the existence of COVID genome, and this is very rapid test. So all these are the things which are because of the advancement only we are able to find out. Otherwise, it would have gone very difficult to test out the causes and to test the material. Now, this is not the hidden thing. So, because of that only, we are safer to some extent, because early diagnosis, early treatment, early vaccines, so vaccination. So, all these things are supporting to fight the corona 19 or COVID-19 coronavirus. So this is how the techniques comes to our rescue, modern techniques. And one more thing here I wanted to focus, that was about COVID-19, I said. But as far as our analysis of some phytoconstituents are concerned, these techniques were rarely seen, or any very big institution used to have advanced instruments. Even the UV spectrometer was rare to get. Electrophoresis, it was not common. Then if you want to, botany student want to perform electrophoresis, he has to run biochemistry department. That is the situation in university, at university level I am talking about, during our days. Now every lab will be having its own independent electrophoresis unit, and not only 1, 2, 3, 4, 5. DNA electrophoresis, also protein electrophoresis units. It is very commonly used. Earlier those things were not there, and we have to find out, we have to ask professors of other departments, we have to take the permission, and then we have to go to other department. They will perform. You never know what is the technique they have done it, and others will perform and they will get you gel. So that was the scenario during those days. Now everybody knows that what is gel, what is agarose, what are the bands, how there is a migration, and how the separation of DNA takes place, or a separation of proteins takes place. So everything is known nowadays. Similar way, the spectrometric analysis also tremendously has gone under change. So these techniques, earlier the staining part was there. Now that is no more in the existence. Chromatographically, the paper chromatography comes into the existence. Then the reagents also been developed. The mixture of reagents, we started using the mixture of reagents. Of course, chromatography is, we cannot say it is much advanced, because those days also the chromatographic techniques were conducted and they were done, they were in a practice. But further techniques of chromatography, like two-dimensional ion exchange chromatography, liquid chromatography, thin layer chromatography, different types of chromatography, the people and the analytical chemistry people that started developing, and then their analysis also, or their utility also, people are started taking for the determination of quantitative status of the particular compound. Now colorimetry was the old matter. Colorimetry was the old matter during those days. Now colorimeter is rarely seen in the laboratory, but for some experiment is needed, so it is maintained for that. So that is true. But for main purpose, for the quantitative analysis, this instrument was very important. It has got it was very important during those days. Nowadays that importance has been lost. So nobody goes for colorimetry. Spectrometry, of course, it is there. Now this absorption spectrometry has comes into the picture. Then emission spectrometry is there. Scattering spectroscopy is there. So what way you wanted to percolate the nature of the parameter I'm talking about, not only parameter, the nature of the parameter. Now that means if the parameter is that the nanoparticle is synthesized and its size is like this. So this is the characterization with respect to the size of nanoparticle. But then how that size has been reduced to that extent, what probably might have happened during the reduction of this metal to that extent to get that particular size. These are the nature, or this we can say this is the pattern or the mechanism by which that particular parameter has come into the existence. So people have started looking towards that parameter. So with that direction, and if we need to know, then you need to have those facilities or equipments, and now these equipments are easily available. May not be every instruments with us, but some other institutions, there are some so costly instruments where if the government itself is not permitting, if it is located at this particular place, then from 15 kilometer away from this distance only it should be placed. Otherwise, within the vicinity of 15 kilometer, that same instruments will not be installed. It is so costly, 2 crores, 3 crores, like that, only for research activities. Everybody knows it. So, but then those instruments are available and those can be used. They are easily available. Only you have to work under somebody who is experts or in charge, but then the results can be generated. So this is the scenario. Then physical parameters also, I am not that much willing to take a name of all these things because these are known to you, but they are representing the advancement in the today's research, like fluorescence spectroscopy, X-ray diffraction, then atomic emission spectrometry. Metal studies can be done very easily without any dilemma and without any fluctuation. Everything can be surfaced out. Plasma emission spectroscopy is there. The infrared spectroscopy is there. NMR spectroscopy is there, which is very interesting for us, especially biological people, because to find out the structuring of the organic compounds or secondary metabolites, we go for this, we use it. Then there is spectrometry, that is FTIR, that is not mentioned here, but then even we can analyze the group of the chemicals or nature of the chemicals, or especially chemical groups of the compound, which is very, very important to determine its characteristics and applications if that compound is extracted from the plant. So those things can be also studied. And beside that, now we have some other techniques, like in ecological science, what we are studying, there are all these kinds, especially algorithmic models, ecological footprinting, laser scanning. There are some many things. Of course, this list includes some of the things which are commonly been used also from many days, but there are few things which are, as a matter of advancement, people are studying, ecologists are studying and coming to the inferences, and those inferences are very, very useful for the determination of strategies and policies of government. That also I would like to put up here. So earlier the taxonomical aspects, now we have the renowned taxonomist here, Dhabe sir. Probably he may be able to discuss these points more better way than me, but just I wanted to say that, sir, during those days, we had limitations in terms of, first limitation was getting the plant for proper identification. Unless you go and locate it, and unless you go and collect it, unless you see the flower or leaf arrangement of that plant, you will not be able to find out, to put up its botanical name properly, which will not be otherwise matching with the flora which is given or which is scripted. Now this has become very easy, but during those days it was very difficult to go. And then the organizing long tours for botanical student was the best for this only, to see the flora and collect the plant. See the flora and collect the plant. Not only angiospermic plant, but fungal materials and also bryophytic, pteridophytic plants, etc., etc. Almost all. So the field work was very, very important during those days. Then maintenance, their maintenance, if some things are, some plants are been collected, they need to maintain, and the herbarium has to be prepared. But then, if now the situation is like that, if you have a sample of any part of the plant, you can go through its molecular modeling, you can go its genetical aspects. In support of that, you can go for its cytotaxonomical concepts, you can go for chemotaxonomical concepts, and you can reach to the conclusion that probably this plant is approaching this particular group, or it comes under that group, because these are the advanced parameters, or it is supported with the advanced parameters. So therefore, these advanced parameters and taxonomies also they are using, and they are very fruitful, they are very useful also. So same kind of things are used in other aspects, not only taxonomy, different other aspects also. In plant breeding, new techniques have come. Now plant breeding basically very rarely been done, but in a crop or cereal, especially it is the agricultural practice. So in agricultural farmhouse, you will definitely getting this plant breeding experimentation and this particular research activities. But now it is same thing, a transfer of characters, transfer of desired characters from one variety to other variety can be done by modern techniques, and those modern techniques are more pronounced, less time consuming, and promisingly yielding the result with specific ratio, and that is appreciated. Only thing is that because they are, when we talk about the modern techniques, then we are approaching the cytological components or genetical components of that particular experimental system. And whenever we are approaching to that particular system, then naturally we are playing with the normal pattern of genome of that particular plant. And when we are playing with the normal pattern of the genome of that particular plant, it is possible that the genome may become unstable. And if it becomes unstable, if it becomes unstable, there is no guarantee it will revert back. And if it revert back, then it is pseudo variant, we can say, or it is those changes or whatever changes the plant will say out of the instability of that genome, then those are the they may not be fruitful for us. So whenever advanced techniques are applied for the transfer of desired character, then it is always associated with the transfer of undesirable character. This is bound to happen because many desirable characters are associated with other characters. So that is one particular term is there, no? Polymorphism or something. So one gene is linked with other gene. So one gene is getting mutated means possibly it may change the it may change the other sequence of other gene also, and that change may not be desirable for you. It may be harmful. So this is there. So some demerits are of course also there. So that cannot be ignored. So that, but then it doesn't reduce the value of that advancement in the advanced techniques in the research. We have to rectify and get the things done. So this is overall impression of different techniques, and especially my intention was not to make you aware about the name of the techniques, but I wanted to just emphasize here that because of the existence of this kind of techniques, the approaches has been changed, and the approaches change, concepts change, and nowadays generation, our research students, they are compatible enough are justifying the research activities at global level. Otherwise, it could not have been appreciated, but it never happens. what you do, what research you do in Institute of Science, what research you do in BRC, what research you are doing in TFIR, what research we are doing in ICT. It's not neglected globally at international platform. It stands for appreciation. And that appreciation we get because we have changed our tendencies, we have changed our strategies, we have changed our approach of working particular concept. And that change in the approach is exclusively because of the um, uh, use of techniques which we started adopting at every level. Now, for this, of course, the researchers, newcomers, teachers, and administrators equally involved. There is a participation, cooperation, synergistic cooperation of all these components. Therefore, this is possible. Otherwise, we could have been at the place where we were there before some days, and all this development could not have been in existence. So, thanking you very much, sir, for giving me this opportunity to share my views. My views were just to provide a guidelines that you really change your approach whenever if you are starting the research anywhere by any means. You should start it with some ideas that are justifying today's need, and for that advancement is available, equipments are available. You take the help of it and get you recognized. That was my intention. Thank you very much, sir.
Thank you so much, sir. It was a very wonderful session, knowing about our past and future advancements in the techniques in our biology. Thank you so much, sir. I would like to ask all the participants, if you have any queries, you can post it on live chat in our YouTube channel. On YouTube, you can post it on live chat. I will read it out to sir, and sir can answer your queries.
Right.
We do not have any queries till now, sir. We shall wait for some minutes for any queries.
No, one thing I would like to add here, madam.
Yes, sir. Yes.
See, when we think of any advanced thing, it is my willingness, or it is your willingness, or the willingness of the researcher. For that, he should not get hesitate that no, I do not know techniques, so why I should go there. That kind of understanding has to be removed, because if we start thinking that way, we will not be developing. So if I do not know something, then it should be this one. In addition, that willingness I should have that to get the information and get expertise skills and get work done. So that kind of things should be there. But we should not underestimate our work. Whatever way we are doing, we should believe that 100%. We should confidently believe that yes, it is correct and proper. That means what the equipments are not there, that cannot be a lacunae.
Yes.
Equipments are not with you. They are somewhere there. So you take the help of it, go there, spend some time and get that knowledge. And but you do not just put that as a minus point for your development. This is, I would like to. And any kind of this one, if you want any advance or any information, I request all the participants to communicate me, and my research students also. One of my research students is already there. Sheetal Sharma is. I could see her. So I think will be helping. As a teacher community, it is our duty to help you all.
Yes, sir, definitely.
Madam, do one thing. Whenever you receive any query, so we can share with the sir, and then after at the end of the program or maybe tomorrow, we can be sorted out their queries.
Fine, sir. Fine.
Yes, sir. Yes, sir.
Okay, so you may proceed for the next lecture.
Okay, sir. Thank you so much, Professor Vijay Mendulkar, sir, for this wonderful session. And now I would like to proceed with our next speaker, but sorry to inform you that our next speaker, Dr. Sanjay Jagtap, who is the Joint Director of Higher Education, Konkan Region, Panvel, will not be present for now due to certain academic urgencies. And also our next speaker, Professor Dr. Avinash Ade, also will not be able to join in due to certain academic urgencies. But we definitely have our next speaker, Dr. Sangeeta Godbole. Let me introduce to you Dr. Sangeeta Godbole, madam. Madam is an associate professor and head of the Department of Botany at Jai Hind College, Churchgate, University of Mumbai. Madam holds a PhD degree and has published many research papers in various national and international journals. She has a specialization in protein purification and characterization. She is a recognized guide for the subject of botany, and under her guidance, a student is pursuing PhD. She has 25 years of teaching as well as research experience. She was awarded Senior Research Fellowship by Department of Atomic Energy, Government of India, awarded Junior Research Fellowship by Department of Atomic Energy, Government of India, and National Merit Scholarship by Government of India. She has been invited for many talks and guest lectures at various organizations, and she is also a very active member of various academic bodies. We are so obliged to have you, ma'am. Ma'am, I request you to commence with your lecture. Over to you, madam.
Thank you, madam. I'm Dr. Sangeeta Godbole, as just mentioned. I'm very happy to be a part of this conference. I thank the organizers and principal, Dr. Rajiv Singh, also for giving me this opportunity to come in contact with so many research people. Forum. Today, I have been an active lecturer, and my topic that I have taken and chosen is more into plant defense. Plants are very close to my heart, and definitely their defense is very important, and therefore this is the topic that is secondary metabolites and plant defense, which I will immediately start. Can I present, sir?
Yeah. Yes, ma'am.
Yeah. So students and my fellow colleagues, everyone on this forum, forgive me if I go a little fast or a little slow. If there is anything that you would like to ask me, interrupt in between, I will be very happy to answer. In this conference, recent advances and opportunities in botany, I'm very happy to present this topic on secondary metabolites and plant defense. To begin with, we all know that plants are surrounded by enormous number of potential enemies. And the fact that plants cannot move, they are sessile, and they have to really defend themselves, be there and defend themselves. If they have to do so, they cannot run and they have to face. The defense that the plant shows is divided into two categories. One is the structural defense and one is the biochemical defense. So if you're planning to do a research in plant defense, you can take any of these or both of these and work on it to find how you can help the plants defend themselves. Although there's a lot to learn from the plants themselves, structural defense to begin with could be cutin, it could be waxes or molecules called as the substance called as suberin. Basically, cutin is nothing but a macromolecular polymer, and waxes are complex mixtures of fatty acid lipids, and they are creating a water repellent surface on the surface of the plant. Suberin is a principal component of the outer wall of underground organs. So whether it is an aerial organ or whether it is an underground organ, the plants have made necessary arrangements to protect itself. As you can see in this picture, you have the section showing you the presence of a cuticle. We always know that it is a cuticularized epidermis is what we say. What is a cuticularized epidermis is nothing but this macromolecular cutin, which is a part of this cuticle along with the surface wax mixed with it. On the top of the cuticle, you may have a layer of only wax, which is not allowing the penetration, the water to accumulate on it, because if water accumulates on the surface, the microorganisms will get water for their multiplication. Also, there is a cuticular layer below the cuticle, which contains even carbohydrates associated with the cutin and the wax layer. So this by itself forms a kind of defense, so which the molecules are, which the pest or the pathogens are facing as soon as they come or encounter a particular plant. Same way with the underground regions of the plant, you have the suberin tissue, which is also, as I said, the part which is associated with cork cells, and the periderm, which is forming even the outer bark of the stem. Coming to secondary metabolites, after the structural defense, we come to molecules which are secondary metabolites. When you begin with this topic, everyone wonders what exactly are secondary metabolites. Are they required or present in all the plants? They are basically a diverse array of organic molecules that initially, when research was on, believed to have no direct role in their growth and development, and they were called as secondary metabolites. And as of now, they are a diverse array of organic compounds, and they do not have any direct role in growth and development, but they do have some role. Actually, it was the organic chemist, which started looking into these molecules because they are often used as medicines. Even in very small amounts, they are used as medicines, and in higher amounts, they can be even poisonous substances. Many of these secondary metabolites are used in the industry as flavoring agents, and so chemists were very interested in these molecules. And it is only after finding their structures, botanists started finding if there are any more secondary metabolites present and if they can be made use of. And a whole lot of research began, with every student, research student trying to find out, is this a plant of my interest? Can that have a molecule which is going to be helping my country or my people who are suffering from different diseases, or the industry who's looking out for different molecules? These molecules also have restricted distribution. Secondary metabolites are not like primary metabolites, carbohydrates, proteins, which are always present in all the plants. They often show restricted distribution to certain families of plants. For example, the alkaloids will be present in the Solanaceae family. Alkaloids are distributed in 20% of the vascular plants. You have the phenolics, you have the terpenes, which are restricted to certain kinds of plants. So that we will see what are the types of secondary metabolites which can be used. So basically the plants, the ability of the plant to defend itself is profoundly affected by the ecological functions also, which are contributed by these secondary metabolites. And the most important function attributed to secondary metabolites in plants is defending them against the herbivores, which continuously try to eat them off, or the pathogens which are infecting them. They can act as pollinators and seed dispersals. Why pollination and seed dispersal is important? If in any area the plant is going to die because of the adverse ecological conditions, if the insect attractants are there to take the seeds away to far off areas, there will be a chance of survival. Agents of plant-plant competition, that is allelopathy, when one plant produces any chemical which is going to hinder the growth of the surrounding plants, it is said to be allelopathy. It could be a beneficial effect also, it could be a lethal effect also, but this plant is making its survival better by creating certain secondary metabolite molecules which is not tolerated by the surrounding plants, and therefore all the soil and the nutrients are available for this particular plant who is producing the secondary metabolite. So one thing needs to be remembered, however, that these secondary metabolites are lethal at times, absolutely lethal to these grazing animals and also mammals. If you are also going to eat those plants, you have to be careful. And therefore, although it may increase the reproductive fitness of the plant, at the same time, it can make the plant undesirable for consumption of the food, and care needs to be taken about that. So coming to directly the types of secondary metabolites, there are three main types of secondary metabolites: the terpenes, the phenolics, and the nitrogen-containing compounds. To tell you about the synthesis of these are the common pathways, which are shikimic acid pathway, malonic acid pathway, mevalonic, and MEP, that is methyl erythritol phosphate pathway. All these pathways, if you see, it is the metabolites present from the carbon metabolism, whether it is TCA cycle or whether it is the glycolytic pathway or it is the pentose phosphate pathway. Metabolites from these pathways are picked up, and these pathways are going to create these important secondary metabolites. So if the plant anytime wants excess of nitrogenous compounds or excess of phenolic compounds, it is going to target the enzymes which are going towards the synthesis of these metabolites, or increase in the transcription of these enzymes which are towards leading to the synthesis of these secondary metabolites, especially if an insect pest is going to be hindered by terpenes. Then the plant will produce more of terpenes by transcribing the enzyme that is going towards the synthesis of metabolites of terpenes through the mevalonic or MEP pathway. Coming to each class, what is a basic molecule? We cannot cover the biosynthetic process, but we must know that terpenes is the largest class, and a basic structure of the terpene is an isoprenoid unit, isopentyl unit, C5 unit. So if you talk about, okay, a plant has a monoterpene, one must know at that level that a monoterpene is two C5 units coming together. So the basic monoterpene geranyl diphosphate, which is formed with the two C5 units, it's a C10 monoterpene. So when I mention a monoterpene, immediately one should know that we should know that monoterpenes means two isoprene units. Diterpene means it's going to be two monoterpenes coming together. So C20 is diterpene, C30 is triterpene, and C40 is tetraterpene. If the carbon atoms go beyond 40, then it is going to be a polyterpene. So basically, with any terpene you're talking about, when you decompose a terpene, what you get is a basic C5 isoprenoid, and that is why they're also called as isoprenoids. And when you have a C5 molecule, isopentyl, added to a geranyl phosphate, you get C15 sesquiterpene. Now, these I have specifically mentioned because monoterpenes, diterpenes, tetraterpenes, triterpenes, and sesquiterpenes, farnesyl, these are the basic molecules which will add further groups and be giving to different kinds of terpenes and different kinds of plants, and therefore you find this as the most diverse class. But the basic unit is C5 isoprene unit. So you have the common terpenes, as I told you, that initially it was always believed that secondary metabolites have no role in the primary metabolism. But if you look at these molecules, you know gibberellins, you know brassicinosteroids, all these plant hormones like abscisic acid, brassinosteroids, gibberellins, all of them are nothing but either a diterpene or a triterpene or a sesquiterpene. Abscisic acid is C15 sesquiterpene. You have certain sterols which are present, which are just derivatives of this triterpene molecule. Just a small change in the structure changes that molecule, and that is the reason we have so many different types of molecules present in so many different types of plants. A slight change in the structure creates a change in the property of the substance. It could suddenly be a molecule of great importance to industry. And therefore, a lot of research needs to be done on this structural aspect of the defense molecules, whether it is terpenes or alkaloids or phenolics. Then you have the polyterpene dolicol, which is also a primary metabolite carrier of sugar in the cell wall, and during that is during glycoprotein synthesis. But do terpenes have anything other than that? Yes, they play a very, very important role as defense molecules. And that is the reason the primary metabolites are kept aside and come to the defense molecules of terpenes. And you all must have heard already about pyrethroids. The insecticide pyrethroid coming from chrysanthemum species is showing very striking insecticidal activity, and it is now sold in the market as pyrethroid insecticide of organic origin. You also have, you can see this glandular hair which is present on the spring sunflower balsamorrhiza, and these are beautiful, very beautiful cases of showing how plants are advertising their toxicity even before the insect has had a trial bite. So you can see that these are nothing but C10 monoterpenes and C15 sesquiterpenes, which are found in the glandular hairs, which are projecting out of the epidermis on the plant surface all over at all times, projecting their toxicity and scaring away the insect pest. And they are synthesized in the same cell of the hair of the epidermis, and this cap is nothing but an extracellular space that is formed when the cuticle and the portion of the cell wall is pulling away from the remainder of the cell. So this is one very good example of monoterpenes which are present in plants. Then there are also, it's not always that you find that these terpenes are produced in the plant after the insect attack, before the insect attack. Sometimes it happens that in case of corn, cotton, and wild tobacco, certain monoterpenes and sesquiterpenes are emitted, and they are produced and emitted only after the insect feeding has begun. So there is some signal after the insect starts feeding on the plant. The signal is given to the plant. We'll come to that signal, how that is there by the signal transduction pathway. But till now, we first talk about what exactly happens with them. As soon as the insect starts feeding on them, the substances repel ovipositing herbivores and attract the natural enemies. So the cotton and the tobacco plant will start producing some kinds of terpenes which are attracting the natural enemies of these feeding insects. And they are asking for help from the other organisms, which could be the parasitic organisms, to these particular insects, and they send these terpenes in the air because they could be volatile terpenes, and they will defense. This volatile terpenes are not only defenses in their own right, but also providing the plants for calling for defensive help. That is what is seen in nature, and plants have the ability to attract natural enemies of plant feeding insects and provide help to themselves. Now, these are the volatile terpenes, which are not essential oils, as you know, are present in peppermint, lemon oil, basil, sage, etc. And this is the basic limonin. When I say monoterpene, a monoterpene is C10. You can see this is a C10 molecule, and this is coming from limonin. And it's a volatile terpene, and it can help. It is used also for protecting the plants. The plant itself is using it. You have menthol, which you all know, which is from peppermint. This is also a monoterpene. Now again, from lemons, you have lemanoids. Now lemanoids are not C10. You can see the structure. They are C30 molecules. So they are triterpenes. And when I say triterpene C30, that means 6 of the isoprene units are present. And they are non-volatile, and they are also anti-herbivore compounds, largely used now as insecticides. You have also as a directin, which is well-known group of triterpenes again, and it's the most deterrent, powerful deterrent to insect feeding and repellents. You also have some molecules of terpenes acting as sterols, because a group of plant sterols have the same basic structure as the insect molting hormone. And ingestion of these plants by which are called as phytoecdysones are the molecules which are present, and ingestion of this plant containing phytoecdysones will definitely disrupt the molting pattern of the insect and affect the developmental processes, and with lethal consequences. These phytoecdysones are seen in the common form Polypodium vulgare. And That was about terpenes, volatile. We did some volatile terpenes, and we did some steroids, and we did some non-volatile terpenes. And we come to the next class, that is the phenolic compounds. Phenolic compounds, if you talk about a basic structure, it's a hydroxyphenyl functional group on an aromatic growth ring. So just an aromatic benzene ring with an OH. That is what is a phenolic compound. It's also a heterogeneous class group, nearly 10,000 individual compounds with a basic structure, which I just mentioned. And they are having their diverse functions. Defense is always there, but they also have function in attracting pollinators, absorbing UV radiations, mechanical support, and reducing growth of nearby plants, that is allelopathy, as I mentioned. And the most important prominent function is defensive against herbivores and pathogens. When you talk about the phenolic compounds, these are the secondary metabolites: simple phenolics, flavonoids, lignin, condensed tannins, miscellaneous phenolics. And all of them, if you can see, they are either formed by the shikimic acid pathway or the malonic acid pathway. Now, the shikimic acid pathway is the major pathway. And if you see, there is a link here between the primary metabolite phenylalanine, which is a common amino acid, and the cinnamic acid. From cinnamic acid begins the course of secondary metabolite. And therefore, the conversion of phenylalanine to cinnamic acid is done by an enzyme which is phenylalanine ammoniolase. And if you want to study whether there is any increase in the phenolics in the plant, all that you need to do is you study this enzyme. Is there any increase in the enzyme activity of phenylalanine ammoniolase, which converts phenylalanine to cinnamic acid? Once there is an infection, you will see that phenylalanine ammoniolase is triggered. The transcription of this enzyme is triggered, and it will start converting phenylalanine to cinnamic acid. And once cinnamic acid is formed, you get a host of all these appendant phenolic compounds, secondary metabolites. May it be benzoic acid, caffeic acid, or coumarins, or lignin precursors, or flavonoids. So all these are basically a slight change in the initial structure of your flavonoid, that is your benzene ring with the hydroxyl group. You come to simple phenolic compounds, which are playing a great diversity of roles in plants. And to begin with, I will tell you about the caffeic acid and ferolic acid, which may be released into the soil by certain plants, and they are affected by the growing of the surrounding plants, that is the allelopathy. And if you try to separate these molecules in the lab, and you try to use caffeic acid and ferolic acid and try to germinate some seedlings in the pot, you might find the effect that it is lethal to germination of many kinds of plants. You may even pick up some plants which are not lethal to these. Possibly those plants have got some mechanism to obstruct the ferolic acid or caffeic acid. And you may use this information also as a target to your research to look into those molecules which are allowing the plant to be not being sensitive to caffeic acid or ferolic acid. Then there are the other flavonoids. You have phenolic compounds which are coming from umbelliferae plants. That is, this is the structure of a simple coumarin. But if a coumarin, you add a furan ring, as in case of furanocoumarins, which are present, soralin is an example of a furanocoumarin molecule, where a furan molecule is added to the coumarin structure, and this soralin is going to exhibit phototoxicity to insect herbivores. Why is it toxic to insect herbivores? Is because this particular soralin, in presence of UVA radiation, after it absorbs the UVA radiation, it's gone into the insect, and it is going to play a havoc by getting itself into the pyrimidine basis of the DNA and affect the transcription and repair of the DNA while it is replicating, and the insect is getting definitely dead with the furanocoumarins gone into its body. Concentration of furanocoumarins, research shows that is increased almost 100-fold in plants like celery after it has been exposed to these furanocoumarins. Then salicylic acid is also a phenolic compound, which is very important growth regulator and is involved in systemic resistance to pathogens. What do you mean by systemic resistance? When there is an attack to a leaf, the whole plant is ready to accept an attack from a pathogen because the plant knows that a pathogen has reached a leaf and it may reach the stem and the roots at any time. And therefore, there is a systemic response exhibited by the entire plant. And who tells the cells of the entire plant that there's going to be an attack? This is told by a molecule of salicylic acid because it's a plant growth regulator and it is also the transmitter of information in various ways to the other parts of the body of the same plant, also to other plants. If it creates methyl salicylate, volatile compounds, then it can even tell the signal, send the signal to surrounding plants that there is an infection that is coming, there is a pathogen that has come, stay alert or start producing defensive molecules is the signal given by the molecule, simple phenolic molecule, salicylic acid. Lignin is also a phenolic compound. You know lignin is a branched polymer of phenyl propanoid group, and it is a very important molecule whose structure is not really known to us. It's very difficult to take out lignin separately because it is very covalently bound to cellulose and other polysaccharides which are present on the cell wall. It strengthens in the normal course the vascular tissues, that is the vessels and tracheids which are present in the conducting tissues. But with respect to its protective functions, physical toughness deters feeding by animals. Chemical durability makes it indigestible to the herbivores and they cannot digest the food that they've eaten because it's very hard to digest this polymer, which is very highly branched polymer. And lignification is also blocking, like a structural defense, it is blocking the growth of pathogens, and it is a common response to infection or wounding. Wherever there is a wound on the plant, the lignin tissue comes there to block the entry of the microorganisms. The last class of phenolics is the flavonoids, not the most. It is one of the most important ones because the flavonoids are again 15 carbons arranged in 2 aromatic rings. So basic phenolic was a benzene ring with an OH group. Here in flavonoids, there are 2 benzene rings you can see connected by a 3-carbon bridge. Now, even if you as botany students don't go into the chemical structure, basic chemical structure you need to understand to find the difference between the different types of flavonoids which are present in plants because their structure changes, their activity and the use also changes. So when you want to design a molecule, if you know something a little bit about the structure, it is always helpful. So as I said, all the flavonoids will contain ring A, ring B connected by a 3-carbon bridge. One ring is coming from the shikimic acid pathway, the other from the malonic acid pathway. So these C6 coming from here, the C6 and C3 coming from the shikimic acid pathway. They are classified, different kinds of flavonoids, thousands of them. They are classified on the basis of the degree of oxidation of the 3-carbon bridge. And many of them will have a glycoside attached at the ring at 3 position here. And many of them are glycosides because they have a sugar moiety attached at this ring. Now, 4 major groups of flavonoids, as you will see the differences, that is flavones, isoflavones, flavonols, and anthocyanin. Now, each of this I will explain to you. The anthocyanins, you know, are colored flavonoids. You can see the structure here. This is the ring A and this is the ring B. When there is a sugar moiety attached to it, this is what is an anthocyanin pigmented flavonoids. And surprisingly, when you get red, pink, purple, blue color of flowers, fruits, vegetables, the leaves that you see, that is because of these anthocyanins which are present. And what makes the color red or blue or pink? It is this hydroxyl group which is present. Increase in the hydroxyl groups on this molecule. If you have too many hydroxyl groups, then the structure changes to in such a way that it absorbs a slightly longer wavelength and the molecule appears more blue, or the plant appears more bluish in color. If the hydroxyl group is replaced by a methoxy group, then the plant appears more red because it absorbs at a lower wavelength. So looking at the structure also, you will know whether the plant is going to be red or blue or green or whatever color. So depending on that, anthocyanins, remember, are the molecules which have a sugar normally attached at position 3. If this sugar is removed, you get anthocyanidin. So the difference between anthocyanin and anthocyanidin is just that anthocyanin is a glycoside and anthocyanin is a glycoside, and when you remove the sugar, the glycon left is what is the anthocyanidin. So the colors are done. And now you come to, after anthocyanins, the second type of flavonoids are flavons and flavonols. You often see that both flavons and flavonols are present in the epidermal layer of the leaves of all green plants, and they generally absorb light strongly in the UVB region, that is 280 to 320, while allowing the radiation of the visible range to pass through uninterrupted for what? For photosynthesis. So if this UVB radiation was not absorbed by these flavons and flavonols, they would have hindered and damaged the cellular macromolecules, because UVB radiation is known to induce mutations in the DNA and will create oxidative stress, and it will damage the cellular macromolecules in the plant. So it is protecting the plant from the UV radiation, because these molecules are absorbing the UV radiation. Then you also have the flavons also in flowers. The insects, remember, can absorb, can see at these wavelengths, whereas we cannot. So if you see this flower, humans see this flower, it is going to appear like this. You can't see the nectar around it. But if the insect sees this flower, then the flavonols can be seen by the insect, and this is called as a nectar guide. So you often see, or at times you don't see also, because they are absorbing light at different wavelengths. You may not see also the symmetrical pattern of stripes, spots, and concentric circles which appear around the nectar areas. These are called as nectar guides, and they are all formed by flavanols attracting insect. They may attract the insect to even trap a feeding insect, but they are insect attractants. Isoflavones. Now, before I tell you anything about isoflavones, which are also called as isoflavonoids, these are important flavonoids with respect to cancer research. These molecules are well targeted for their activity to stop cell division and be effective in treatment of cancer. But since our topic is not that, we are talking about isoflavones. What and how are they different? This was the normal flavon which we were mentioning, the 3-carbon bridge and the 2 structures. You know it was at position 2 that the ring B is attached. In isoflavones, you will note that this ring B is attached at position 3. Immediately the molecule is coming under the isoflavone. Just the change in the position of the second ring B gives so many different properties to this molecule, and it has got a very high pharmacological activity and is found largely in many legumes. And different biological activities that it shows with respect to disease resistance, pest resistance, is the insecticidal activity. We all have heard about rotenone, which is used as an insecticide, a pesticide, and a pesticide, fish poison as well. That is an isoflavone. You also see that this sheep which is grazing on clover, which is rich in isoflavonoids, often suffer from infertility because this isoflavonoid ring structure has a 3-dimensional structure which is similar to a steroid hormone which the insects have, and these allow this particular molecule which has come from the plant to bind to the estrogen receptor in these insects, and they will create a havoc in the insect, and the insects will not be able to grow or even reproduce on plants containing isoflavones. So that is the anti-estrogenic effect seen on the grazing sheep. And then lastly, we have phytoalexins. Now the term phytoalexin, which I'm going to tell you later as well, are any antimicrobial compound. It could be a terpenoid, it could be a phenolic, it could be a glycoside, it could be any, it could be a protein, any compound which is released and helping the plant to take care of the microbial attack that is made. So antimicrobial compounds synthesized in response to any bacterial or fungal invasion, they are all coming under a target that name, that is phytoalexin, and isoflavones are seen to be acting as phytoalexins in controlling the microbial attack, and therefore they are also a type of phytoalexin seen in some plants. The isoflavones, you have the last of the phenolics, that is the tannins, which you all know. They are always toxic, and grazing animals don't really like the food which is rich in tannin, and mammals, they are always a feeding repellent to mammals like cattle, deer, apes, and they avoid food with high tannin content, all these grazing animals. Also, surprisingly, you will see that unripe fruits have got tannin, and this helps the plant. Which way? If tannin content is high in unripe fruit, the animal will not try to pluck it out because it doesn't like the smell of the tannin, and therefore it will allow the fruit to mature. If the fruit matures, then the seed within will also mature, and then the fruit is dispersed to areas and the seed can germinate. If unripe fruits were picked up by these animals, then the seeds would not have been matured, and that would be a loss to the plant. So basically, tannins will be either condensed tannins, which are nothing but the flavonoids polymerizing to each other. You can see all these are the flavonoids which are polymerizing, and they are a constituent to all the woody plants, and they're also giving rise to the anthocyanidins. So they are also called as proanthocyanidins. The second type of tannins are hydrolyzable tannins, which are not polymers of flavonoids only, but they are heterogeneous polymers made up of phenolic acids and also simple sugars, and they're easier to hydrolyze. Dilute acids can hydrolyze them, and they are much smaller than the tannins. But in any case, these are general toxins and reduce the growth of the survival of the growth and survival of the herbivores. Now coming to the last class, that is the nitrogen-containing compounds. They are all metabolites synthesized from the common amino acids, and you have them divided into either the alkaloids, cyanogenic glycosides, glucosinolates, or non-protein amino acids. We'll discuss each one of them. Alkaloids, as you all know, have got a dramatic effect on mammals. And because this is because they affect the central nervous system of all the mammalian organisms. And if you have the humans also consuming the alkaloids in higher amounts, it can cause to death. A large family found, normally this alkaloid is a large family, and about 20% of the vascular plants will find the presence of these alkaloids. They're synthesized from one of the few common amino acids like lysine, tyrosine, tryptophan. These are the precursors for all the alkaloids, and they are seen to be defensive against the predators, especially the mammalian predators. And they have got large amount of medicinal properties. And most important is that when the grazing animals, the domestic grazing animals are fed or are allowed to go for grazing, sometimes surprisingly, they prefer alkaloid containing food, and it results into death of the organism. And large amount of livestock deaths are already caused by consumption of large amounts of alkaloid containing plants such as lupins, and there is a lot of research is going on in creating lupin, which is low in the alkaloid content. But what happens is, if you make a transgenic plant which is low in alkaloid content, it is easily infested by the herbivores and other pests, and therefore that becomes a problem. So what they have, there is research going on in that direction. Coming to pyrrolizidine alkaloid, a very beautiful thing that is seen is the pyrrolizidine alkaloids, the nitrogen oxides, as they are present in the plant in this form, are not at all lethal to anyone. But as soon as this nitrogen oxide, which is pyrrolizidine alkaloid, comes in contact with the digestive tract of the insect, what happens is it gets reduced to tertiary alkaloid. This tertiary alkaloid is extremely dangerous for the insect because it can go in, it can pierce through the cellular walls, and it can kill the insect because it takes over the entire machinery. It's very, very poisonous. But some insects show, surprisingly, the capacity. They will turn it into tertiary alkaloid, and they will again return the tertiary alkaloid back into its nitric oxide form, and it doesn't stop there. When this insect is eaten by its own predator, it is keeping this molecule ready to use it against the other predators. See how system works in the environment. So it is taking some molecule from the plant because the plant wants to protect itself. It is not getting killed by the tertiary alkaloid because it already has some enzyme which can again turn it into the safe nitric oxide. It stores this molecule within itself, and when the predator of this insect comes, then it is using this against that predator, possibly because it will not have this enzyme which can reoxidize it to nitric oxide. And this is what is seen in Tyria jacobi, cinnabar moth. This has got the capacity to reconvert it into non-toxic nitric oxide. So these were about the alkaloids. The second group is more interesting, that is the cyanogenic glycosides. They are also present in the plant. Like this is a cyanogenic glycoside. The example I will give you, there are legumes, grasses and species of rose family which will have cyanogenic glycosides. As the molecule cyanogenic glycoside is not at all harmful. For example, durin is the cyanogenic glycoside present in the sorghum plants. And this is present in the epidermis vacuole of the epidermal cells of sorghum plant. And the enzyme glycosidase is situated in the mesophyll cells in the sorghum plant. So the enzyme and the cyanogenic glycoside is spatially separated into two different compartments. As soon as there is an attack by the insect, the cells are destroyed. The contents of the epidermis and the mesophyll come together, and then the glycosidase will act on the cyanogenic glycoside, remove this sugar moiety, and create this cyanohydrin glycon. And the cyanohydrin glycon, either spontaneously or with hydroxynitrile enzyme, gets converted into a powerful deterrent, that is hydrogen cyanide. As soon as the insect starts eating and the cells are destroyed, hydrogen cyanide is released. Otherwise, hydrogen cyanide is not released because hydrogen cyanide and enzyme are in two different compartments. So this is the beauty of the plant, how it is protecting itself. It is not creating hydrogen cyanide otherwise, unless and until it is affected. Now, these hydrogen cyanides, one must know that the tubers of normally this hydrogen cyanide will cyanohydrin will act as a toxin and inhibit all the metalloproteinases which are present as protein digesting enzymes in the insect gut. Iron containing cytochrome oxidase is also one of the metalloproteinase which is a key respiratory enzyme which can be hindered, and it will bring immediate death of the organism. Tubers of cassava are eaten in African countries because it is a major carbohydrate containing staple food. In many tropical countries, you see them, they are high in cyanogenic glycosides. And these tubers are given a particular treatment. They are roasted, they are dried, and a host of treatments are done before they are consumed. But still you find out in many cases that it leads to death of the individuals. And cyanogenic glycoside containing food has to be properly treated before eating. Sometimes they don't have anything else to eat, and therefore their other nutrients are also less, and that is why the effect is very seriously seen, and many people are dying because of this. And therefore research is going on to try to see that the tubers will not have, the tubers are ones which are eaten. The tubers should have less amount of cyanogen glycoside transcribed protein that is present there. The transcription to form cyanogen glycoside via pathways is reduced in the tubers, and at the same time the plant should not be infected by the pest. So the quantity of cyanogen glycoside in the leaves is kept high. So that is one kind which is there. Glucosinolates, exactly like cyanogen glycosides, these are different molecules which are spatially separated and kept ready. Now, glucosinolate is found in brassicaceae. When you cut cabbage and you get a particular smell, you see you don't want to eat it. That is what is the smell which is coming from the glucosinolates being hydrolyzed, found basically in brassicaceae and related families. Hydrolyzed products function in defense, herbivore toxins, and feeding repellents. The glucosinolates are not normally broken down in intact plants. But what do you find in cabbage butterfly? The cabbage butterfly has got the efficiency again to like food which is high in glucosinolate levels, and these levels in the leaf, even if high amounts are present, these can still provide a good residue. You can still you can have these glucosinolates serve as stimulants for adult feeding and egg laying, and isothiocyanate produced after hydrolysis will act as volatile attractants to other cabbage butterfly to tell them, okay, come, here is the food, you can come. So sometimes you do find certain pest organisms which are going to be the ones which are going to help them. So glucosinolate under normal conditions, if the plant has glucosinolate, thioglucosidase is the enzyme which will remove the glucose moiety, and this glycon is then spontaneously converted into isothiocyanate. Okay, and these are and nitriles are released, and hydrolysis of glucosinolates to mustard. It is also called as mustard smelling volatiles, which are going to protect the plants. Lastly, in the alkaloids, you have the non-protein amino acids. Now, you all know that it is the proteins which are formed from a chain of amino acids by linking by the peptide bonds. So the insect requires food, proteins. It is breaking down into amino acids, and then it is utilizing those amino acids to make its own proteins. What happens is when the plants are eaten by these insects, the food that the plant has, the food that has been eaten by the insect contains certain free amino acids. And now these amino acids are not actually the normal amino acids which can be taken in the proteins. They are slightly different. For example, cannabidiol, which is mistakenly incorporated into proteins and result in a non-functional protein after ingestion by herbivores. So what is happening is cannabidiol, instead of arginine, wherever arginine has to go into the protein, cannabidiol goes and sits there. And if cannabidiol goes and sits there, the structure definitely is different. You can see so similar cannabidiol and arginine. The only difference is there is an O that is present here in cannabidiol and which is not present in arginine. You also have proline, which is present, which is very similar to acetylene 2 carboxylic acid, which is its non-protein amino acid. So wherever proline needs to go, by mistake, acetylene can be incorporated. And if the wrong amino acid is incorporated, what happens? The protein structure changes. If the protein structure changes, the function of the protein is disrupted. And if the function of the protein is disrupted, the plant, the insect cannot survive. So these were the 4 types of the nitrogen containing substances. Finally, we come to the plant defense categories. Now, the plant has got structural defense. The plant has got secondary metabolite as defense. How is the plant going to work towards these molecules and using these molecules? Either the plant will always have a constitutive defense response. That is, like for example, nicotine is always present in Nicotiana tabacum, but it can also be induced to produce more of Nicotiana tabacum after there is an insect attack. So molecules which are always present for defense are said to be constitutive defense. It's a constitutive defense response because one type of insect always comes to attack this plant, and to tackle it, nicotine should be always present. So that is a constitutive defense. And most secondary metabolites as defense are constitutive, and they are produced in some plant or the other. Induced defense responses are very interesting. They are occurring or induced to be present only after the damage occurs by the insect, pest, or the pathogen. That means this has to be very, very quick. In order to be effective, the induced response has to be very quick, because by the time the plant produces the response, it should not be totally eaten away by the feeding animal. So let us see what are the constitutive induced defenses. Let us see how the induced defense can be done quickly. First of all, we come to the categories of insect herbivores, that is phloem feeders, cell content feeders, chewing insects. Now, these are the types of feeders like aphids, whiteflies, causing little damage to the epidermis or the mesophyll cells. Cell content feeders, which are just piercing directly and causing intermittent damage, and chewing insects, which can kill the plant in just one night. These are the caterpillars. Voracious feeders cause most significant damage to the plant. So if it is induced kind of defense, it has to be definitely very fast. So let us see how this fast defense comes. Now, this is one example where the plants can recognize specific components of the insect saliva. Now, the plant has to know which insect has come, insect 1, 2, or 3. Depending on which insect has come, is it an insect which cannot tolerate phenolics, or is it an insect which cannot tolerate alkaloids, or is it an insect which cannot tolerate terpenoids? Accordingly, the plant is going to take action. So how does the plant know which insect it has attacked? Now, the insect, when it is eating the plant, the saliva of the insect has some amino acids. So an amino acid called as glutamine, for example, in one of the studies, is seen to be acting with the fatty acid, linolenic acid, which is a content of the membrane of the plant tissue which the insect has eaten. So imagine an insect is eating the plant tissue, and the glutamine amino acid is mixing with the linolenic acid, fatty acid, which is present in the plant tissue. And while it is feeding, there is a conjugate that is formed between the linoleic acid, which is plant derived, and the glutamine, which is insect amino acid. And this conjugate, whatever is prepared, this particular fatty acid amino acid conjugate will form a molecule which is called as an elicitor because it is identified by the plant. Immediately when it sees, okay, glutamine has mixed from the insect, I know which is this insect, and the plant will start its defensive action towards this particular insect. For example, volicitin is one of the elicitor molecules which is seen, which is recognized from the insect saliva, from the signal transduction. And as soon as the voracious eater has started eating, the plant is activated and it can have an induced response. Now, there is another kind of response that is the proposed systemic signaling pathway. Now, whenever there is an attack, suppose this is a phloem parenchyma cell which is being attacked, it will start producing a protein which is called as procystemin. So as soon as the tomato, this is seen in tomato plants, the wounded leaf cell is going to produce procystemin, which is nothing but a large precursor molecule. And this in the phloem parenchyma cell itself gets cut off or proteolytically digested to produce a smaller peptide, which is cysteamine. Now, this cysteamine comes out of the cell and comes in the region, which is intercellular region, the apoplast region, and moves towards the companion cell, which is joining it. Now, in the companion cell, which is joining it on the membrane, there is a leucine-rich receptor, which is always recepting molecules. Is there any cysteamine around? Is there any infection? So the leucine-rich receptor immediately notices the cysteamine and it binds to the cysteamine. The leucine-rich receptor cysteamine complex then gets phosphorylated and it's activated now. What will this phosphorylated leucine-rich receptor cysteamine do? It will go and activate this another membrane protein, that is phospholipase A2, which is the key signaling molecule. The phospholipase A2 is phosphorylated again by this ATP leucine-rich receptor cysteamine molecule, and it sends the signal to the DNA for converting linolenic acid into jasmonic acid. Can you see? PLA2 generates the signal that has initiated the set of reactions which are converting linolenic acid into jasmonic acid in just this companion cell. Once the companion cell jasmonic acid is synthesized, it will be sent to the sieve element. Once it goes, jasmonic acid goes to the sieve element, in no time it is given. The signal is sent to all the parts of the body because phloem sieve cell interconnects the entire system of the plant, and in all the cells of the body of the plant, jasmonic acid is present. What does this jasmonic acid do? It activates the genes encoding proteinase inhibitors in all the cells where it has reached. Now, proteinase inhibitors, I'm coming to that proteinase inhibitors a little later, but let me tell you how jasmonic acid was synthesized in that cell, that is from in the chloroplast of this particular cell. This pathway is taking place where linolenic acid converted into 12-oxo-phytidinoic acid, and then it moves from the chloroplast to the peroxisome, where the enzymes of the beta oxidation pathway will convert it into the final jasmonic acid, and from the peroxisome it will come into the cytosol. From the cytosol it will go into the sieve element, and jasmonic acid will travel to the entire body of the plant through the phloem tissue. What is the use of jasmonic acid being present in all the cells? You will see here, I said it activates the genes. So you will see that jasmonic acid is activated by first linking to one amino acid in that cell, that is isoleucine in this case. So jasmonic acid signaling involves first binding to an amino acid, isoleucine, to form an isoleucine jasmonic acid conjugate molecule. This molecule then is going to join SCF-CoI1 protein complex. This is the SCO1-COF complex to which this complex is bound. Now, this is a jasmonic acid isoleucine SCF-CoI1 complex. What does this complex do? Can you see there is a JAS repressor sitting on this promoter site? What is the function of this JAS? The JAS does not allow at ordinary times the promoter to function and start the transcription of these genes. So So the genes which are maybe for proteinase inhibitors or secondary metabolite in producing enzymes, all these defense proteins which could be produced if the transcription starts is stopped by the presence of JAS attack being present here. So you have the JAS, which is the repressor protein attacked by this complex, and it is removing the JAS, repressing JAS. As soon as it removes the JAS, it throws away the JAS, and JAS is degraded. And this whole thing is degraded. And now who is present here on the promoter side? It is MYC2, which are the transcription factors. They are allowed to bind to the promoter and start the transcription of genes that encode defensive proteins. So this is how, when you say jasmonic acid, so what did we see? There was a signaling molecule which was present, that was your cysteamine, which came and gave the signal to phospholipase, and it is going phospholipase A2, and phospholipase gave the signal to the cell to produce jasmonic acid from linolenic acid, and jasmonic acid then was shifted to all the parts of the body of the plant through the phloem, and jasmonic acid triggered the activity of the genes encoding the defensive protein by removing the JAS repressor and allowing the transcription factors to bind to the promoter and start the transcription of this gene, and the story ends, and all the cells have got enough kind of secondary metabolites which we just saw, or any other kind of molecules. When I talk about any other kind of molecules, I talk about proteins that are inhibiting herbivore digestion. Now what are these? They could be alpha amylase inhibitors. So we spoke about the secondary metabolites which are phenolics and all. Now we're talking about proteins. Even proteins can be. What are these proteins? They could be either alpha amylase inhibitors or proteinase inhibitors. What do you mean by that? An alpha amylase enzyme is present in the insect, and it is digesting the starch that is present in the food that it is eating. But if alpha amylase enzyme is not allowed to act, it cannot digest the starch, and therefore when the food, the leaves are eaten by the insect along with the starch, inhibitor is also there, alpha amylase inhibitor is there, so the insect will not be able to digest starch. It will not be able to digest, it will not eat. We don't eat something which we find it difficult to digest. Same way, when the leaves proteins are coming, the insect proteinases are trying to eat cleave the protein into amino acids by using their proteinases like trypsin, chymotrypsin, or whatever proteinase the insect has. But if the leaf along with the protein has proteinase inhibitors, then the trypsin, chymotrypsin, and all these enzymes which are present in the insect gut, they cannot digest the proteins, and therefore the food that is eaten is of no use. So if there are too many proteinase inhibitors, if there are too many amylase inhibitors, the food eaten has no value, not only for them, for us also. Remember that. Remember, for us, I've just mentioned it, although it's not of any significance, but proteinase inhibitors are produced and they are present so much in all the food, but they are sulfur-rich amino acids. And if you cook and eat them, then they are very, very nutritious proteins. But don't eat them raw. That can create a problem in your stomach. The third type is the lectin proteins. Now, what are these lectins? Are nothing but they bind to carbohydrate containing protein. So as soon as the insect eats the lectin containing food, in its gut, there is the carbohydrate containing protein which binds to the lectin, and this complex binds to the epidermal cells of the gut. And what do? They do not allow any absorption of nutrients from the food that they have eaten, the insect has eaten. And therefore, if lectins are binding to the epithelial cells, they are interfering with nutrition absorption in the insect, and it is of absolutely no use. And therefore, these molecules, jasmonic acid has induced plant proteins to inhibit herbivore digestion. Now, phytoalexin, as I mentioned, I will tell you a little later, is nothing but any chemically diverse group of secondary metabolites with very strong antimicrobial activity that is accumulating in the site of infection. So we were talking till now, we were talking about especially the insect pest and the herbivores, the bigger animals, but at times there is attack from microbes. There could be the microorganisms, like you have the viruses and the bacteria and the nematodes that are the ones which, and the fungal pests which are attacking. So what is the plant doing to survive these attacks? So phytoalexins are compounds that are synthesized by certain plants which can take care of these microbial pests which are present. And these phytoalexins are chemically diverse group of secondary metabolites with very strong antimicrobial activity that can accumulate around the site of infection. Okay, so wherever there is infection, those cells will start producing the phytoalexins. Common mechanisms are there, but different plant families employ different types of secondary metabolites as phytoalexins. For example, you have some family producing the isoflavonoids, or you have some family producing the terpenoids as phytoalexins. So isoflavonoids in the legumes and sesquiterpenes in solanaceous plants are seen to be produced in to combat the microbial pest. And generally, these attacks are induced responses. They are not detectable in the plants before infection. So you can even try infecting a particular plant with microbe and check in that particular plant which kind of phytoalexin is present. A lot of research can be done in this way because we have so many, we have a very rich flora, and if you try to do with an unknown plant, a totally new plant, and try to infect with a common microorganism and check whether if it is going to survive, is it producing any phytoalexin compound, can you find out that phytoalexin what it is? A lot of research can be done on this aspect, and these are regulated by at a transcriptional level only. That is, immediately the genes coding for phytoalexins will be transcribed to form these phytoalexins. So they are also seen in defense, are demonstrated by mutants, as well as overexpression in plants is done for phytoalexin compounds, and they seem to be more pest resistant. Now, these are some of the common phytoalexins that are present. Medicarpine from alfalfa, which is isoflavonoid. Immediately when you see the attachment here in the ring B is in this position, it is an isoflavonoid. You can see this is an isoflavonoid. Again, the ring B is attached at position 3 here in this molecule, which contains the hydroxyl groups and the phenyl ring. And here you can see the isopentyl ring, which is also attached. So it's a mixture of a phenolic compound to which an isopentyl terpene basic isoprenanoid is also attached. So it can be any molecule which is attacking. This glyciolin is from soybean, which acts to protect soybean plants from microbial infection. And then you have the rishutin, which is present here from potato. It is a sesquiterpene. You can see 6C6, C6 plus 3C, so C15. So C15s you all know are sesquiterpenes. So anytime you see a molecule, you must just see what type of terpene or what type of flavonoid molecule that is. Or capsidol, this is also a C15 from that is pepper, and from tobacco. You can see these phytoalexins. Now, when a microbe infects a plant, we are coming to like what exactly happens there. The microbe may be surrounded by your cells which are dead. This is also a hypersensitive response, that is rapid activation of defense reactions in the region where the small microbe has attacked in association with the host cell death. If there is a tissue that is here, the microbe has attacked, you can see the surrounding tissue is going on dying. But when the surrounding tissue dies, it is isolating this microorganism from the rest of the tissue of the plant and protecting the plant. This is what is called as an hypersensitive response, and it is often preceded by some things and some reactions, which is rapid accumulation of reactive oxygen species and the nitric oxide, because both of these are required for activation or making these cells die or forming the hypersensitive response. And this much region may just scrape and fall off, and the rest of the plant is happily surviving. But what are the rapid accumulation of reactive oxygen species you see before hypersensitive response takes place? This is a very beautiful illustration. There is a pathogen, a small microbe which is attacking. It has sent its elicitor, recognizing molecule. The plant has a receptor molecule which is identifying this particular elicitor. As soon as the receptor targets the elicitor, what does the receptor do? It activates the NADPH oxidase present on the surface membrane, and it converts molecular oxygen into a host of reactive oxygen species and brings about a host of reactions, that is a lot of NADPH oxidase dependent reactive oxygen species, that is superanion oxygen and hydrogen peroxide and hydroxyl ion. The most powerful oxidant is created. At the same time, the cell wall proteins, proline-rich proteins, are cross-linking and making the attack or invasion reaction by this organism very difficult, because cross-linking polymers with proline-containing proteins is going to increase, and it will become difficult for it to enter. At the same time, calcium channels are opening, and calcium channel, because of excess of calcium, it is definitely allowing the nitric oxide synthase gene to be activated to produce nitric oxide, which is a secondary messenger. Now, the presence of nitric oxide and the presence of the reactive oxygen species, both of them are required, and it creates this hypersensitive response where the cells are dying, because both of them, this reactive oxygen species itself, is going to bring about lipid peroxidation, then enzyme degradation, enzyme inactivation, and nucleic acid degradation in these cells. And the nitric oxide, along with the reactive oxygen species, will produce the hypersensitive response, that is the death of the cells. It will also at the same time prepare phytoalexins. You know what are phytoalexins? So it will go and it will allow the transcription of genes which are towards phytoalexin synthesis. It will also bring about more synthesis of lignin polymer, so that the cell wall is made more polymerized, and there is difficulty in penetration. Structural defense is what is made ready. And there is salicylic acid biosynthesis as well, which is the molecule which is going to help all the parts of the body of the plant to be intimated that there is an invasion in one area. And biosynthesis of hydrolytic enzymes, that is these enzymes like chitinases, glucanases, and hydrolytic enzymes, which are going to be dissolving the walls of these fungal cells which contain chitin, or the bacterial cells which may be present. They are trying to create hydrolytic enzymes, secrete hydrolytic enzymes, and not allow the pathogen to enter at this level. And because of the salicylic acid biosynthesis, the systemic acquired resistance is acquired, because the message is sent to all the cells of the body of the plant that there is an infection of a pathogen coming at this leaf, and you all have to be ready. So these are the host of reactions which are suddenly taken in whenever a pathogen attacks. And therefore, it could be immediate response in the invader cell. It could be a local response, that is gene activation and accumulation of molecules like your benzoic acid, salicylic acid, and also production of ethylene and production of jasmonic acid, and alterations in the secondary pathway to produce a particular kind of secondary metabolite. Or it could be a systemic response where and gene activation to produce enzymes which are towards breaking of the fungal or bacterial cell wall, that is chitinases, and synthesis also of the pathogen related proteins. And then this is the systemic acquired resistance, which I just mentioned. When a plant has survived a challenge from the pathogen eliciting this information, necrotic lesions you can see all over. And then you can see the SAR, systemic acquired resistance, becoming established throughout the plant like this. The initial pathogen infection was here, and it has given the entire plant the resistance to further attack. That is by accumulation of salicylic acid because of just one leaf infection, synthesis of certain volatile methyl salicylate, because the volatile components are going up into the air and telling the different parts of the body of this plant as well as the surrounding plant that there is an infection, so you all be ready. That is the signal, volatile signal sent. Transmission of the signal to other parts via vascular system is also doing, that is via phloem, and resulting in an increased systemic resistance to pathogen, and airborne transmission of the signal to other parts of the body as well as the neighboring plants. They are all alert. So this is what the plants are doing when there is a pathogenic attack. And finally, the secondary metabolites we have summarized. We have seen the toxicity repellency, we have seen the structural defense, and finally we have seen the biochemical defense, that is due to secondary metabolites or production of signals, reactive oxygen species or nitric oxide or salicylic acid, jasmonic acid or ethylene is synthesized. Induction of the signal, transduction networks, and the biosynthesis of these molecules like phytolexin, lignin, hydrolytic enzymes via signal transduction network. So that is the end of our plant defense, and I hope you all have gained some knowledge from what I have just spoken. If there are any questions, I'm free to take them.
I request the students, if you have any queries, post it. Yes, we already have some questions, ma'am. I will read
Yes.
it out to you.
Sure.
The question is, how to identify plant species for bioactive studies? What are the general or ecological characteristics of plant species we have to observe for selection of plant species for bioactive studies?
Yeah, very good question. Because we have such a wide flora, the first thing would be a literature survey to find out which plants are already worked upon. Now, many a times when you have some study which is already available where plants are worked upon, you get a system how this search can be done. But if you read properly articles and on research, you can find the ways in which you have to study. But the plant of your interest could be if you are trying 10 or 4 or 5 plants selecting. And if it is a plant defense, the topic on which you want to study, then you can try actually stressing the plant with the metabolites and checking out if the plants have got some kind of metabolites which are of interest to you. So there is no single one rule that you have this plant or that plant. But if you can also go to grandma's pouch and find that there are some plants which are showing some phytoalexin kind of activity, antimicrobial activity. There are so many plants which are producing phytoalexins. Phytoalexin can be any compound. When it is showing you antimicrobial activity, it is being produced. And if it is being produced, if you want to study the phytoalexin activity, you can select those plants which are already known and which are not worked upon with respect to the molecules. Now, I would also like to mention that when you are working on the plants, it's not just the plant molecule, the plant showing insecticidal or phytoalexin activity or any defense activity. You have to reach to the end to find out what that molecule is. And can you create an artificial molecule like that? Should also be your focus. If it's a medicinal molecule, whether you can create a molecule like that. So plants you can select by initially trying a few experiments on trying to deliberately stressing the plants and finding if there is any change. You may even do a polyacrylamide gel electrophoresis to find the proteins which are stress related proteins if you're working on proteins specifically, because when the plant is not stressed, have a protein profile, and when it is stressed, you get another protein profile. A second. You get another protein profile, and therefore it is definitely one of the ways in which you can find out the methods. If you want to study that whether phenolics are increased, you can also do the same thing, estimate the total phenolics, the total terpenoids, or by normal methods that you all have after stressing the plant with the desirable pest or the microbial species which you want to test on, and then select the plants accordingly.
Okay, thank you, ma'am, for answering this query. We are waiting for other query. Yes, the next question is, how futuristic it is working with biochar as a tool for plant disease management?
I didn't get the question properly. Can you please repeat?
How futuristic it is working with biochar as a tool for plant disease management?
That needs to be worked on. I think other dignitaries who are present in this meet can also answer if they have better answers, because I don't have a very good answer for your question here.
Okay, ma'am. Ma'am, we also have other questions.
Yeah.
The next question is role of flavonoids.
Yes. Role of flavonoids. Yes. What is the question? Do you want to know the role?
Yeah, just the role of flavonoids.
Oh, I'm glad you want to know the role of flavonoids. Flavonoids are basically, I'm giving you in the summary here, flavonoids are very important molecules. And to say that what the flavonoids can be, I have already mentioned to you that they are very important molecules as insect pollinators and seed dispersals, because they have color pigments with them. They also absorb the UV-B radiation and therefore help the plant do photosynthesis properly and protect the plants from the harmful effects of the UV-B radiation. One use of flavonoids, isoflavonoid, and also they are nectar guides, as I mentioned to you. Then they are also having anti-cancer properties, isoflavonoid group, especially where the ring B is attached at this region. It definitely has anti-cancer property. They have insecticidal, very strong insecticidal activity. You already have rotenone with you. You have anti-estrogenic effect because some of the steroid structures of these isoflavonoids are mimicking the hormones of the insects, and they are creating disturbance in the fertility of these grazing sheep, because the three-dimensional structure of the steroid is definitely targeting the estrogen hormone and binding to it and causing problems. Plus, isoflavonoids, which are also flavonoids, are having a very strong antimicrobial activity. So depending on which plant is producing which isoflavonoid, you have the phytoalexins of that particular plant. You can study them. And finally, you also have tannins, which are flavonoids and which are feeding deterrents, can be used in for protecting the plants, the herbivorous plants, the larger mammalian plants. So these are all the uses that I just mentioned about the flavonoids, in case you missed.
Okay, ma'am, thank you. The next question, our next question is there any ecological indicators for selection of plant species?
That was the first question that was also asked, which I just mentioned. The ecological indicator would be, you can try to see, you should always do research which is application oriented. So if you can just try to find out, are there any crops which are infested by a particular pest? Are we finding it difficult to take care of these pests? Try to work on areas. In India, we face so many problems in agriculture. If you can focus on what are the present problems in agriculture and select your plant or your research, it will be of use to someone, and it will be of use to our own country. And therefore, you should always have proper literature survey of what is required, and you should do research in that direction. That is what I would say. So ecological means it always depends on where the need is. Where are the pests and where are the pests controlled? We don't require to do anything more. Which are the pests which are not being controlled by all the available forms of control? Whether you increase the secondary metabolite in the plant, because a lot of things are there. If you increase the secondary metabolites in the plant in a particular area, and if the plant is a crop plant which is eaten, then you have to take care that the secondary metabolite is not creating disharmony with your system. So you require then the secondary metabolite should be expressed only in the part of the plant which is not eaten by humans, if it's a crop plant, and it will at the same time be increased in the part of the plant which is only infested by the pest. So I would suggest that you try to survey in agriculture what are the problems that are faced, because this is a continuous evolution. Remember, the plants are getting evolved and the pests are also getting evolved. So as you have a resistant variety, slowly you will also have a resistant pest. So why is that pest resistant? What has changed? What has it done? Like, for example, I gave you some examples in case of alkaloids. I told you about this particular Tyria jacobi silver moth insect pest. It has got the ability to again reconvert it back into the nitrogen oxides. So are the pests having such ability? Have they been able to do that? Then is there anything else that this silver fly can be made or can be tackled? So you try to find out what is the need in agriculture in whichever ecological zone it may be. So try to go in that application-oriented research is what would be my advice.
Okay, ma'am. Thank you, ma'am. And ma'am, just one last question I would like to ask you before we end this session. That is, which are the best methods used for isolation and purification of plant pathogenic fungi and bacteria?
When you talk about bacteria, then you use the nutrient agar mediums of different types which you are using. And when you are using fungal, then you can take help of a microbiologist, because the fungal media are slight friend, right? So you will be requiring, whenever there is an infection to a plant, you have to scrape that particular region and you have to then culture. And then you have to work on what kind of organism. You have to identify that organism, find whatever is available information on that organism. So when you're talking about how you isolate, the normal procedures which are used for isolating any microbial organism, whether it is a bacteria, you use a set of media. When it's a fungal, then you use the potato dextrose medium and all that. Those normal are the ways in which you will identify. And there are many research institutes which are only working on there may be people who are only working on the fungal contaminants that are there of certain plant species, if you're talking about plant species, or there could be some institutes which are working on certain types of bacterial pathogens which are there on plants. So the methods remain the same. The initial methods, as Sir said in the morning, that the basic research is always going to remain the same. And you have to then talk about the other methods which are the further finding more about the organism is up to you. I hope I have answered the question and asked the question.
Yes, ma'am. Ma'am, thank you so much for this wonderful session. With this, I would like to end this technical session 1, and I would officially take this opportunity to thank our keynote speaker, Professor Vijay D. Mendulkar, for accepting our invitation and being an important part of this conference and sharing his knowledge with us. I thank you also, ma'am, Dr. Sangeeta Godbole, for accepting our invitation and sharing your valuable time and knowledge with us. Also, I would like to thank Dr. Sanjay S. Jagtap and Professor Avinash Ade, although they couldn't be present here today, but accepting our invitation is a great thing for us. So, with this, I would end this session. Thank you, sir. Thank you, ma'am. And we will meet again on the same platform for our technical session 2, which will begin at 2:30 p.m. And I would request the participants to join a few minutes earlier. Thank you all. See you again shortly.
Thank you.
Thank you.
Thank you, everyone.
Thank you, Sangeeta ma'am. Thank you.
Thank you, sir. Thank you so much.
त्याला क्रिकेट सांगून ठेवा ना. आवाज बंद करायचा ना. OBS. आपण दोन टाकून डेस्कटॉप कशाला ठेवायचं. ठीक आहे. कारण हा जो घेतोय ना, हा जीमेटचा घेत असणार ना डेस्कटॉप. झालं. युट्यूबला तेच दिसणार, इकडे पिन करणार. ऑडिओ एक, दोन, तीन.
तीन मार्क केले का?
Good afternoon, everyone. Welcome to our technical session 2. We shall start with the paper presentations now. To begin with, I call upon the first presenter, Mr. Santosh Shippar. Mr. Santosh Shippar, are you present?
Yes, I am getting your voice. Yes, I will start my presentation just.
Okay, sir, you can start your presentation.
Yeah. Okay, am I audible?
Yes, sir. Yes, you are audible.
Okay. So, good afternoon all. My presentation topic is Anther culture in Indian brinjal cultivars. Okay. So, under this title, we will discuss the different points, that is introduction, objectives, need and applications parameters used for the development of parental lines or Anther culture. Then protocol flowchart, then basics, observations, result and discussion, troubleshooting, future perspectives and references. Now, by using anther culture, we can produce the doubled haploids. Doubled haploids are nothing but the parental lines which are homozygous, and from these homozygous lines we can develop the different hybrids. Okay, so haploids which are produced from the male gametes or female gametes. That is the haploids are diploidized spontaneously or artificially in a laboratory, that is in vitro. Okay, and different techniques we can use here for the development of such double haploids, parental lines, or we can say homozygous lines by using androgenesis, gynogenesis, and gamma irradiation. Okay, so the anther culture, why we select? Because this technique is a more promising and convenient for the manipulation. Okay, and in this anther culture, or by using this anther culture technique, we develop different parental lines. And why we selected the brinjal? Why we selected the brinjal? Because brinjal is a widely cultivated crop plant in all over world. And as Mendel selected pea plant for its for his experiments, for the anther culture we can select the brinjal because lot of variations we can get related to the morphological characteristics. For example, in case of fruit, different shapes, sizes, colors, spiny and non-spiny nature we can get from the brinjal plants. In case of leaf, we can get narrow, broad, hairiness, or spiny, non-spiny characteristics. In taste also, brinjal is different. In different region, taste will be different. Okay, so and the brinjal can be cultivated in all seasons. So for initiation of anthers, we will have the buds in all seasons. Now, anther culture is used for the development of new pure lines. This is our objective. Okay, so development of new pure lines, double haploid parental lines or homozygous lines from commercial hybrids through anther culture with different traits as we discussed. Now, what is the need of double haploid brinjal? By using this technique, we have to develop disease resistant lines. Means these brinjals which are developed by this technique should be resistant to the bacterial wilt. Then to develop new lines with different shape, size and color, to develop new lines for higher yield. Then we can save time. We can save time which is consumed during the breeding, and 100% homozygous or pure lines can be developed by using this technique. And now here on this slide you can see the comparison between double haploids and conventional breeding. Means by conventional breeding we can develop several parents. Okay, several parents in 6 to 8 generations. Means 6 to 8 generation we have to spend, and for the completion of 6 to 8 generation of these plants we will take 3 to 4 years. Here you can see the 100% heterozygous plant we will have in the form of F1 hybrid. Now after selfing, that is in F2 generation, we will get heterozygosity and homozygosity in 50-50% ratio. Okay, in 50-50 ratio. So in F3, F4, F5, F6, F7, and later on in F8 we will get the homozygous parental line. And this parental line which is developed by conventional breeding will achieve 99.2% homozygosity. But in case of double haploid technology, from F1 hybrid we can get pure line in only 1 generation, and time needed is 8 to 12 months. Means we can save time from 3 to 4 years to 8 to 12 months, and 100% homozygous plant or 100% pure plants we can get from the double haploid. Now here for the androgenesis or for the development of double haploids we used 4 F1 hybrids from the different seed companies, that is Utkarsh, Utkal, Ajay and Kirti. So among all these hybrids, Utkarsh is for high yielding, Utkal is for disease resistance, that is bacterial wilt, Ajay for taste, and Kirti was for earliness. Now what type of parameters used for the developing such lines? Okay, so the buds we have to collect from the donor plants, and these donor plants are grown in the controlled condition, that is in the growth chambers. And these donor plants which are grown should be healthy and free from the different microorganisms. So if we grown donor plants in the healthy environment or in the closed growth chambers by providing external air which is filtered, then the donor plant will be healthy. And from these donor plants we have to collect flower buds. We have to collect flower buds which are having different sizes and sepal petiole The anther color and microspore stage is another third parameter that in that greenish yellow anthers we have to use, and in these greenish yellow anthers we have to use late uninucleated or early binucleated microspore for the embryogenesis. The nutrient media which is used for this is BRCI1, which is supplemented with 5 mg per liter kinetin, 5 mg per liter 2,4-D, and 0.2 mg per liter vitamin B12, which is additives, and the sucrose concentration is 12%, which is induction medium, and regeneration medium, that is BRR1, which is supplemented with 0.1 mg per liter kinetin and 3% sucrose. Now, we are well known from the sporophytic pathway. So we have to inhibit that sporophytic pathway into the gametophytic pathway. And for the conversion of sporophytic pathway into the gametophytic pathway, we have to treat these buds or anthers with different temperatures, as we have to provide pre or post treatment with 4 degrees Celsius or 35 degrees Celsius. Now this is the flowchart of protocol which we had used for the development of pure lines. Now here we have to collect buds from the donor plants, and these buds are sterilized with the 70% ethanol for 1 minute and 5% NaOCl for 15 minutes. After each treatment with the sterilant, we have to use sterilized distilled water for washing thrice. Then these anthers, which are isolated from sterilized buds, are kept on the induction medium, that is BRCI1, for 8 days in dark at 35 degrees Celsius. And after 8 days, we have to keep these anthers or these cultures in a dark for 4 days at 25 degrees Celsius. Means total for induction medium or the anthers kept in induction medium for 12 days. After 12 days, there is a subculturing, and these anthers we have to keep on the regeneration medium, that is BRR1, at 25 degrees Celsius in dark. After every 3 weeks, we have to subculture. After 1 month to 4 months, we will get embryos from these anthers. And after achieving the 4 to 5 millimeter embryo height, or if plantlets develop, these plantlets we have to keep in MS medium for the further growth. After 20 to 25 days, we will get well developed plantlet, and this plantlet we have to acclimatize in the greenhouses, that is primary hardening, and after primary hardening we have to do secondary hardening after 10 to 15 days. Means acclimatization phase is needed here for the plantlets which are developed in vitro. Now here you can see the colorful pictures from which you can understand better the anther culture in the commercial brinjal hybrids. Okay, so here is the donor plant. Here is the donor plant, and from these donor plant we collected the buds. Now the proper size of bud we have to select for getting the greenish yellow anthers. Then this anther we have to isolate and inoculate in the BRCI1 medium, then BRR1 medium. We will get embryo from these anthers, and these embryos will be converted into plantlets, and plantlets will form the haploid plants or double haploid plants which are developed spontaneously. Now the haploid plants or double haploid plants means putativs. These putativs we have to test for ploidy. Now with ploidy analyzer we can analyze the plant is haploid or double haploid. Now if the plant is double haploid, we can say the parental line or homozygous line is developed from the anther culture, and the trial will be carried out, and the trial will be carried out in the field. If haploid plant ratio is more than the double haploid plant developed, then we can use the several chemicals, that is colchicine or azalene, for the diploidization of these haploid plants. Here you can see the haploid brinjal plant developmental stages. Excuse me, my screen is visible?
Yes, sir, your screen is visible.
Okay. So haploid brinjal plant developmental stages. Here on the screen you can see the donor growth conditions. In A or first photograph, you can see the how donor plants are grown in healthy conditions with fan pair system of the polyhouses. Then these donor plants grown in healthy conditions will produce the different buds with good vigor. These buds we select on the basis of sepal petal ratio or their sizes. Then we isolate anthers. Then we have to test the stages of different microspores, whether they are uninucleated or binucleated. Then from these anthers we have to produce embryos, means embryo germination, then and then finally plantlet development. From the developed lines we can see here the plants are having different leaf morphology, this variant leaf morphology among new lines, then variant plant morphology among the pollens. Okay, so normal pollen plant we had got, then abnormal plant also showing abnormal pollens, then normal pollen germination and abnormal pollen germination. We have grown the pollens from the haploid and double haploid plants, or we have germinated. And if suppose we have the germinated pollen grains, then we can say the plant from which pollens collected is a normal plant, means the developed a pure lines. If abnormal pollens are there, no germination on the germination medium, then we can say this plant which is producing such pollens is a haploid plant. Okay. Then here you can see the plant spininess. First plant is without spines and second plant is having more spines. This is the variation. Now here you can see the plant morphology, means if normal morphology of plant is there, then the pollens after staining we will say that is the normal pollens, and with the normal morphology plants or with normal pollens, the normal fruit setting we will get. So this is the double haploid. And if abnormal morphology of plant is there, if we collected pollens and stained, then we will get the abnormal morphology of that pollens, these pollens. So abnormal morphology of plant, abnormal pollens, and fruit setting is abnormal. And these seeds are mostly without, these fruits are mostly without the seeds. Now as we are discussing different points or different we are using different hybrids for the anther culture. Among these 4 hybrids, Ajay hybrid is showing the different parental lines with the different fruit colors. Okay, so here you can see DH1, DH2, DH3, DH4, DH5, DH6, DH7 and DH8. These are the samples only, because we had produced the lot of lines from the Ajay F1 hybrid, which was the heterozygous, and these all lines around the donor fruit are homozygous. Now here you can see the double haploid plant fruit with control. With control means the Utkarsh fruit is from the donor plant, which is the heterozygous. And from this heterozygous, we had developed the homozygous lines. Okay, see here is the clear-cut difference in the color of the fruits. Now here Utkarsh fruit, which is from the donor, showing more shiny. Here is the less shiny and color also faint. Then here is the dark color and more shiny than the donor plant. Okay, so such type of differences in case of Utkarsh we got. Then in case of Utkal, the see here the Utkal fruit is having different morphology than the parental lines developed. Here also Kirti is having a greenish color and whitish color on the fruit, but after the developing or after the development of parental lines, we got the different that is the parrot green color of the fruit. Okay, then whitish, which is different from the donor. Now here this slide will show the trial in which we used the 2020 plants for the testing of homozygosity. Result and conclusion. Here we had initiated number of anthers from these 4 hybrids, that is from Utkarsh. We had initiated 2,472 anthers. Then we got 53 embryos, 40 plantlets, 38 plants we hardened, and out of these 38, we got the 7 plants which were of pure line. Okay, now out of 38, 31 was haploid. Okay, so same in case of Utkal, Ajay and Kirti, but in case of Ajay, we had initiated 6,448 anthers, out of which we got 391 embryos, 338 plantlets, then 296 plants hardened, and out of these 296 plants, 275 was in the secondary hardening due to mortality, and from these 275 plants, we collected seeds from the 59 plants, because 59 plants was pure lines, and the remaining 216 plants or remaining other than the 59 plants were the haploid plants. Here with the graph we can get the comparative analysis of anther culture in 4 F1 hybrid. The first was Utkarsh, second was Utkal, third one was the Ajay, and fourth one was the Kirti. And among these 4 hybrids, the more responsive F1 hybrid was Ajay. Summary we can get here. Among 4 hybrids, Ajay shown the more or highest DH response, which was good considered, and 0.91% DH was formed from the 6,448 anthers. So F1 hybrid is promising and highly responsive for anther culture protocol. Okay, why? Because F1 hybrids are more vigorous. Okay, then from results or from observations and result, it was concluded that we need to improve protocol for other hybrids which are poor responding. Means for Utkarsh, Utkal and Kirti, we have to develop new protocol for getting good DH response. Now we had faced troubleshooting, that is the mortality of anthers was means percentage of mortality of anthers was more. Then the embryogenesis response in case of anther culture was inconsistent. Then the fruit setting percentage was also significantly low, and seedless fruit also we got, which was not having any need in the further generation. Then optimization of future perspective, that we got approximately 75% haploid plants and 25% double haploid plants. We needed only 25%, means 75% were waste. So for the utilization of haploid plants, we treated these 75% haploid plantlets with the colchicine. And this is our also future perspective for the developing haploid plant into the double haploids. Now, here we can see in case of anther cultures, experiments of anther culture was tried initially, in which response was good in genotype Ajay, but in other there was low response. So optimization of anther culture protocol for them is necessary. Here we are also using shade microspore culture, alternative to the anther culture, and normal plantlet formation ratio was low. Now in this plate, in second plate, you can see one plantlet is developed among 10 embryos. Among 10 embryos, we will have only one plantlet. In case of anther culture, among 10 embryos, we can get 4 to 5 plantlets. Okay. Then in such shade microspore culture, we have to refine the protocol. Microspore culture is tedious, so more efforts needed to perform the microspore culture. And these are the references. Okay, thank you. The topic is open for discussion. If any questions are there, you can ask.
If anyone has any queries, you can ask.
Hello, Sangeeta ma'am. If you have any
Yes.
question,
Yeah. Can you hear me?
yeah.
My first question to Mr. Santosh Appar is that how much time did you require to do this work, all this work?
Actually, for getting double haploid plant, we spent approximately 1 year.
1 year, okay.
1 year. Okay, yeah.
Okay. And you have used colchicine and all that?
Actually, for a trial, we had used not in large quantity, but just shoots we had treated with colchicine.
I also want to know whether simultaneously the plant breeders are also working on homozygosity by plant methods for these plants.
Yeah.
Are you in touch with the plant breeders also?
Actually, we can share parental lines with the plant breeders, and
Okay.
they can be hybrids, which are the promising. Okay, we can save their time for the development of parental lines. We can get parental lines in 1 year.
Yeah.
They will 3 to 4 years for the development of parental lines.
Okay. That's good. Excellent work that you have done, and you have managed to do this also. I'm sure a lot of field and good lab would have requirement would have been there for this to do this work.
Yeah.
So you've got support from your college, which is this college?
Badrinarayan Barwali Mahavidyalay, Jalna.
Okay. Good. Excellent work that you have done. I'm very happy to hear about all this that is done.
Thank
nicely.
you. Okay.
Yeah. Thank you.
Oh. Any other questions? Yeah. Open for discussion. Yeah.
Mr. Santosh Ipar, I want to inform you that someone asked on YouTube, Mr. Prakash Patil asked you about role of flavonoids. Can you elaborate his question? What is the role of flavonoids? On YouTube live chat, he asked you. You might be don't know about him.
Role of flavonoids.
Flavonoids on live chat.
Good afternoon, sir.
Yes, sir.
That question is earlier sessions.
Okay.
Yes.
I answered to that question of role of flavonoids. It was my.
Okay.
Actually, topic is different, sir. Actually,
that was question was in our chat box actually.
Yeah. Okay.
From the earlier session that was answered over. Yeah.
Okay. Thank you.
Okay, so if we do not have any, we can move on to our next presenter. Shall we? Yes, I think we should move on to our next presenter. Thank you, Mr. Santosh Ipar.
Okay, yeah.
And now I call our next presenter, Mr. Viraj Chapke. Mr. Viraj, are you present? Mr. Viraj, are you present?
Everyone, Viraj is not presently. So, our next presenter.
Okay, so Mr. Viraj is not present. So we can move on to our next presentation. I call upon Ms. Dipanvita Bhushal for the next presentation. Ms. Dipanvita.
Hello. Yeah, I'm present.
Yes. Ms. Dipanvita, you can start with your presentation.
Yeah, sure. Just a second. I'm just sharing my screen. Hello. Hello.
Yeah, it's been shared. It's been shared.
Yeah. Can you see my?
Yes.
Yes.
Yeah.
Yes, it is visible.
Okay. Good afternoon, everyone. My topic is basically the HPTLC quantification and estimation of rutin and camphorol using the aerial parts of Ixora javanica and Ixora barbata. I will cover the introduction, material, methods, result, and discussion, conclusion, and references. Okay. As we all know that our world is actually relying on medicines. We are already relying on plants for the medicines. So it has been seen that the World Health Organization estimates that 80% of the people in developing countries of the world are basically very much dependent on this primary medicines for their healthcare, and about 85% of traditional medicines are extracted from plants itself. So plant secondary metabolites constitute very important source of drugs to be used for their medicinal values. Plant flavonoids, as discussed by Gogole ma'am, which are a group of phenolic compounds and are of very much important classes of secondary metabolites. Flavonoids are present in aerial parts of every plant species, and it is widely distributed throughout the plant kingdom. And it is known that up till date, the 300 varieties of flavonoids are there. They are actually very much in high demand for their medicinal purpose, and thus the quality control and qualitative analysis of these phenolic compounds in traditional herbal medicines have become a necessary undertaking. These phenolic compounds have a very broad range of physiological activities such as anti-cancer, antioxidant, anti-inflammatory, antibacterial activity, etc. So it is the hour of need that phytochemical evaluation is one of the tools where the quality assessment it includes preliminary phytochemical screening, chemo profiling, and marker compound analysis using modern analytical techniques. And a modern analytical techniques, as our Mendulkar sir in the morning session, as he discussed, there are very much advanced techniques where we can actually we can lesser our time for the sample extraction and utilize it to get the result in very good time consuming factor will be very less for us, and we will get a highly authenticated or accurate result from it. So in the past 2 decades, HPTLC, which is high performance thin layer chromatography method, has emerged as an important tool for the qualitative as well as quantitative phytochemical analysis of these medicinal drugs and also for its formulation. The major advantage of this HPTLC is that several samples can be analyzed simultaneously, which requires only small quantity of mobile phase. HPTLC is also suitable for estimation of chemical constituents present in the plant materials. Same quantification of quercetin and rutin by HPTLC method is also reported in Elephantus scaber. And I have seen, I have done the survey where I have seen that there is no report available on the quantitative studies of the phenolic compounds of the selected plants that have selected Ixora javanica and barbata. So with an aim to know what are the phytochemical present in this plant, I have done the simultaneous extraction of rutin and camphorol in the leaf and flower extracts of Ixora javanica and barbata. The plants that I have selected, that is Ixora javanica and Ixora barbata. Ixora javanica is an evergreen spreading shrub which is native to Java. That's why its name is Ixora javanica. Its leaves are opposite, inflorescence terminal, style equal. Flowers of these plants contain anti-cancer, inflammatory, antioxidant properties. That is the main reason that I have selected this plant for flavonoids to study. In Ixora barbata, it is a glabrous shrub introduced in Kolkata garden before Roxburgh's time, but its native country until Kurz found it in Andamans. It is also known as bearded Ixora because barbata in Latin means bearded, and it refers to the woolly mouth of the corolla. Here the leaves, morphological characters are leaves are opposite, inflorescence are terminal, corolla tube long, stamens 5. Commonly it is cultivated in Calcutta and elsewhere in India. Phytochemical studies of some species of Ixora coccinea, Ixora finlaysoniana, Ixora arborea, etc. indicates the presence of important phytoconstituents such as lupeol, ursolic acid, oleanolic acid, cystosterol, rutin, anthocyanins, glycosides of camphorol. So the literature survey that I have done indicated that Ixora javanica and barbata were not yet analyzed by the modern technique of HPTLC. Therefore, I just selected this plant to study the constituents. Here, this is the plant. This is the plant. My first plant, that is Ixora javanica. You can see the leaves and flowers. The freshly leaves and flowers were collected from the Acharya Jagdish Chandra Bose Indian Botanical Garden, BSI, Calcutta. There itself, the plant was authenticated. Then I have taken the leaves and flowers where it was rinsed and with tap water and I have again washed it with distilled water to remove the dust, pathogens, whatever it was there. The standard compound rutin and camphorol was procured from Sigma, and the mobile phases that I've used in the study were of SD fine and the HPLC grade petroleum ether, 85% methanol and 95% ethanol water were of HPLC grade. This is the next plant. This is Ixora barbata, the leaves and the flower. This is the way I have shade dried my leaves and flowers, and this is the campus of Acharya Jagdish Bose from where I have collected my plant sample. Now, the material and methods. The collected plant specimen, the leaves and flowers, as I said, was separated, washed and dried in shade. Then the dried material was powdered in a grinder and it was stored in the amber colored bottle. From that, 25 grams of the powder of the plants, that is the leaf and flower, was taken in the thimble of the Soxhlet extractor. 250 ml of, now here, this extraction that I have done is a consecutive extraction. So first I have taken 250 ml of petroleum ether, which is of HPLC grade, and and done the extraction for 4 hours to get rid of the lipids and fats. Then after 4 hours, the concentrated petroleum ether which I got, I just filtered it through Fortsman filter paper number 1, and a little was kept aside for the phytochemical screening. And the remaining extract which was there, it was concentrated in the rotary evaporator till I get a semi-solid mass. This was then further run on an HPTLC. Now, the defatted powdered plant material which was placed in the thimble was kept for drying overnight, and it was again extracted with 250 ml of 85% methanol for 8 hours. This released free flavonoids and glycosides in the methanolic solvent. This concentrated methanolic extract was again same way, same procedure that I've filtered with Wattsman filter paper number 1, and the little was kept aside for to check the phytochemical preliminary screening, and the remaining was evaporated in the rotary evaporator till a semi-solid mass was obtained, and for the running of to run in the HPTLC analysis. And this was labeled as 85% methanolic extract. So I've got petroleum ether extract. This is second 85% methanolic extract. And then again, continuing with the extraction process, I again I have taken that powdered plant material, which was again placed in the thimble. It was dried again, and then it was again placed in the extraction Soxhlet extractor for with 250 ml of 90% ethanol for 8 hours again. And the same way I've again filtered it with the Wattsman filter paper number 1, cooled it, and then taken the little portion for preliminary test, and the remaining evaporated in the rotary evaporator, and to keep it to run it in the HPTLC analysis. This was labeled 90% ethanolic extract. Thus, these leaves and flowers extracted in all the 3 solvents using above extraction method are finally subjected to HPTLC screening. Similar HPTLC screening was also reported in the leaf extract of Syzygium cumini. This is where I've done my Soxhlet extraction in my college. You can see the Soxhlet extractor. These are the way I've filtered, and this is my concentrated solution after I've got from Soxhlet extractor. And here, this is the rotary evaporator that I've concentrated more my plant extract till I get a semi-solid mass. Now, we move on to the, as I've kept, previously I've said that I've kept a small portion to check the phytochemical screening. So this is what first I've done. I've checked the phytochemical screening. Second, I have done is the determination of total flavonoid content by aluminum chloride calorimetry assay. In this, I have prepared a standard curve where routine standard was taken by dissolving 10 mg routine in 10 ml methanol. Then I have measured the aliquots accurately, ranging from 50 to 350 μg per ml, and transferred to different test tubes. To these different test tubes, I have added 4 ml of distilled water, and then I have again added 0.3 ml of 5% sodium nitrite. And after 5 minutes, I've again added 0.3 ml of 10% aluminum chloride. This was mixed thoroughly. Then after 5 minutes, again I've added 2 ml of 1 molar sodium hydroxide, and also I've made that volume up till 10 ml with the distilled water. Now this, all the test tubes that I've made, it was kept for incubation for 30 minutes at room temperature. The absorbance of the standard solution was measured in a spectrophotometer at the wavelength 510 nm, and it was read against the blank. Same way I've prepared my sample, and that sample I've taken was 1 mg, that semi-solid mass that I've got from the rotary evaporator of all the 3 extract, that is petroleum ether, 85% methanol and 90% ethanol. From that, I have taken 1 ml of respective solvents, and wherein then I have added in different test tubes, and from that I've added in the centrifuger for 15 minutes in 10,000 RPM and filtered. The filtrate that I've got were taken in the fresh test tube, 20 ml test tubes. To this, I have also added distilled water, 4 ml of distilled water. Now, this is the same procedure that I have done for preparation of standard curve. Same chemicals I have used, same range, and it was measured at 510 wavelength. The total flavonoid content was expressed as mg per gram of the extract. This is the phytochemical screening of the leaf extract of Ixora javanica and barbata, and the table number 2 shows screening of the flower of Ixora javanica and barbata. As you all can see, I have done test for alkaloids, flavonoids, saponins, tannins, and this is plus, triple plus, or double plus is less, moderate, or high turbidity that I've got while doing my screening, and negative is that I've not got any turbidity in the test tubes. The main part is the separation of flavonoid by HPTLC method. First, the preparation of the sample. from the semi-solid concentrated mass, that is petroleum ether 85% methanol 90% ethanol. From that I have weighed 20 mg of the plant material and dissolved in 1 ml of HPLC grade petroleum ether, respectively 85% methanol and 95% ethanol, and sonicated for 5 minutes. The standard compound rutin and camphorol, which was procured from Sigma and Merck, and the mobile phases in the present study, it was, I just spoke about it earlier, it was of SD fine. Now, the standard solution and linearity. The standard stock solution of routine was freshly prepared by dissolving 5 mg of standard compound powder in 5 ml of methanol and camphorol, respectively. And it was sonicated for 5 minutes. From this stock, 7 different concentrations were made, that is from 100 to 700 μg per ml. The linearity of each of these standard was determined by applying standard solution of different concentration ranging from 0.5 to 3 μg per spot. All the solvents used in this analysis was of HPLC grade. Now the chromatographic condition, it was the plate was of silica gel 60 F254 HPTLC plate, which was 20 into 10 centimeter. Both the samples and standard 10 ml each was applied in that. And next we go into detection and quantification of the compound. Here also you can see the TLC plates what I have used in that routine and camphorol was seen in 250 nm and 366 nm wavelengths using that CAMAC TLC scanner, and the software was used was WinCat software, and the RF and AUC dilution factor, all the things where I got the result immediately. Second comes the development, chromatogram development, the mobile phase that was used. It was ethyl acetate, acetic acid, formic acid and water in the ratios 100:11:11:27. It was kept for 30 minutes at room temperature. Photo documentation was done. The instrument itself, it does everything. Then the plates were derivatized. Next comes the result and discussion. From the phytochemical screening, I have seen that that is from table number 1 and 2. This table number 1 and 2, I have seen that there was present in my plants, that is Ixora javanica and barbata, leaf and flower extracts in petroleum ether 85% methanolic and 90% ethanolic solvent, showed me the presence of alkaloids, flavonoids, terpenoids, glycosides, phenols, saponins, etc. The present study thus provides evidence that the solvent extracts of these selected plants contains medicinally important bioactive compounds, and this thus justifies the use of the above plant in traditional medicine and for treatment of various diseases. From the total flavonoid content of the plant, by aluminum chloride calorimetric assay, a calibration curve was plotted. As you can see in figure number 3, it was observed that petroleum ether extract gave better result, followed by 90% of ethanolic extract for Ixora javanica, whereas here you can see as in for petroleum ether gave better result, followed by 90% ethanolic extract, whereas 85% methanolic extract gave better result, followed by petroleum ether for Ixora barbata. 85% methanolic extract gave better result, followed by petroleum ether. This is the comparative study that I found out. The highest amount of flavonoid content was found in the flower extract of Ixora javanica in petroleum ether, which is 330.7 mg per gram, followed by the flower extract of Ixora barbata, which is 300 mg per gram. And the least was obtained at in the solvent 85% methanolic extract, leaf extract of Ixora javanica. Now, routine and camphorol content in the plant extract of Ixora javanica and Ixora barbata. The calibration curve was prepared by plotting the concentration of rutin and camphorol versus average area of the peak, and its linearity was in the range between 50 to 250 μg per ml, as you can see in the figure 4 and 5. The correlation coefficient of rutin was found to be 0.96 and 0.97. For rutin it was 0.96 and for camphorol it was 0.97. The after derivatizing the plate, when it was scanned at 366 nm, rutin and camphorol showed single peaks, which peak in HPLC chromatogram at 0.45 and 0.67 RF respectively, which is here figure number 6 and figure number 7. The experiment was performed in triplicate for reproducibility accuracy. The obtained data was analyzed statistically. The result obtained of rutin and camphorol contained in the plant extract of leaf and flower of Ixora javanica and barbata in petroleum ether and 85% methanol and 90% ethanolic solvent are discussed below. Now in this table number 4 and 5. The table number 4, here we can see the leaf and flower extracts of Ixora javanica and barbata using petroleum ether extract, 85% methanolic extract and 90% ethanolic extract, indicates that the rutin and camphorol are present in both the plants. Very high amounts of rutin was extracted from leaves. Very high amount of rutin was extracted from both the leaves as well as flower of the plants using 85% methanol as the extractive solvent as compared to petroleum ether. The highest amount of rutin was observed in Ixora javanica flower in 85% methanolic extract, followed by the leaf extract of Ixora barbata in 85% methanolic extract. However, very low amounts of rutin was found in you can see here it is 90% ethanolic extract of Ixora barbata. In table number 5, we can see that camphorol was found in Ixora javanica and barbata in petroleum ether extract and 90% ethanolic extract. Very high amount of camphorol was found in 90% ethanolic extract of the leaf part, and followed by the 90% ethanolic extract flower part of Ixora barbata, and the least was observed in 58, which is 90% ethanolic extract of leaf of Ixora barbata. I did not get any result in 85% methanolic extract. Therefore, it was suggested that camphorol extraction is best in 90% ethanolic extract followed by petroleum ether extract. Based on the above observation, we suggest the use of 90% ethanol as preferred solvent followed by petroleum ether for extraction of camphorol, and avoid the use of 85% methanol. And for routine, the 85% methanolic extract is the best solvent to be used. This is the table, comparative table. I've made the bar chart of it. It is as same as this. I've just made it in bar graph. This is the photo plate that is which was seen under UV 366 nm. Now we move on to the conclusion. This paper is the first to cite the simultaneous quantification of rutin and camphorol compounds in the extracts of Ixora javanica and Ixora barbata. The densitometric estimation HPLC method used for the simultaneous quantitative estimation of two flavonoids, rutin and camphorol, in leaf and flower extracts of Ixora javanica and barbata, it was found to be an efficient method to screen leaf and flower samples in order to assess its quality and authenticity. Therefore, the proposed HPTLC method can be used as an analytical tool for quality evaluation of plants and formulation containing rutin and camphorol as chemical markers. Phytochemical investigation of leaf and flower extracts of the above two plants also revealed the presence of many other bioactive compounds such as alkaloid, terpenoids, saponins, etc. in addition to flavonoids. Here in this plant we can also, I mean, it is very, I have got very good bioactive compounds, and I have left it for further researchers to do more research on these plants. As both the above plants showed presence of higher amounts of flavonoids, this plant is suggested to be used as a potential source of natural flavonoids to exploit their antioxidant, anti-tumor and anti-inflammatory properties. And our work suggests that use of 85% methanol as the best extractive solvent while extracting rutin from both leaves as well as flowers of Ixora javanica and Ixora barbata when it is compared to extraction using 90% ethanol or petroleum ether. However, for extraction of bioactive compound of camphorol, our work suggests that use of 90% ethanol should be preferred, followed by petroleum ether, and avoid use of 85% methanol as the extractive solvent, as no significant presence of bioactive constituent camphorol was observed in both leaves as well as flowers of both the plants. Now, the information derived from this is left for further researchers to work on extraction to see if there are more metabolites present in the plant. Lastly, I would really thank the authors. We authors are really thankful to Institute of Science, Mumbai. We personally thank Professor Mendulkar sir and Dr. Sopan Kharat for providing their kind help for sample analysis. Thank you. I am really thankful to Dr. Sopan for giving me this opportunity to present my presentation today. Thank you. Any questions?
Madam, there is one question.
Yes.
Hello.
Hello.
Am I audible?
Yes, yes. Am I audible? Yes.
Madam, first of all, your presentation was very nice.
Thank you.
But I have one query. Whenever you told in case of Soxhlex extraction,
yes,
Soxhlex extraction, at that time you told you use the 250 ml of petroleum ether.
Yes.
So, what is the capacity of that thimble?
Thimble. In thimble, we just put. I have thimble. Therein I have put the powdered part. That is the leaf and flower, which is 25 grams. The solvent that is of 5. That depends on the capacity of the bowl that you buy. The Soxhlex thing. It may be 500, it may be 1000 ml. I used 500 in that.
Praveen sir. I am clearing your question. I am also involved in this research. So that time we use 500 ml thimble.
Okay, sir.
500 ml thimble we use that time.
And sorry, there might be some mistake because I am presenting for the first time.
No, madam. No. Actually, I am confused, madam. So I am asking that question.
No.
Don't worry. Yes.
I'm saying I was presenting.
Actually, we take a 2/3 solvent related with that particular bowl. 2/3 हम लेते हैं उस bowl का. इस हिसाब से मैंने पूछा. Otherwise, your presentation and your work was very nice.
Thank you.
No problem.
Thank you, Deepanita.
Thank you, sir.
Anybody other?
I have one question.
Yes. Hello. 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 Hello.
Hello, do we have any more queries? We do not have any more queries. Then I think we can move on to our next presenter. Ms. Dipanvita, thank you so much.
Thank you.
And now let's move on to our next presenter. Our next presenter is Mr. Praveendar Shah. I request Mr. Praveendar Shah. To begin this presentation. Is Mr. Praveendar Shah present?
Praveendar, start your presentation. Hello.
Hello.
Yes, you are audible. Start your presentation.
Yes, sir. You can. So just a minute, sir. Network issue. Hello. Screen. Hello.
Start directly.
Screen visible. Screen is visible.
Yes.
My topic is in situ growth of silver nanoparticle in polyvinyl alcohol thin film matrix using aloe vera leaf extract. Its characterization, antimicrobial and biocompatibility study. Next, nanoparticle. The particle having at least one dimension in the range of 1 to 100 meters, such particles are said to be a nanoparticle. So, according to the dimension, there are different types of nanomaterial. Like we have the 0 dimension, 1 dimension, 2 dimension, and 3 dimension. In 0 dimension nanomaterial, all the dimensions are measured within the nanoscale. No dimension are here. It is very compulsory in 0 dimension. The material size must be in the range of 100 nanometers, not more than 100 nanometers. Such type of nanomaterial are said to be a 0 dimension nanomaterial. The example is nanoparticle. In 1 dimension nanomaterial, 1 dimension is outside the nanoscale. Example, nanotube and nanorod, etc. In the 2 dimension nanomaterial, 2 dimension are outside the nanoscale. Example is plate shape like graphene, nanofilm, and the nanolayer, etc. In the case of 3 dimension nanomaterial, the material that are not confined to the nanoscale in any dimension, the class containing bulk powder, bundle of nanowires and nanotube, etc. In present study, what I had taken, we have grown the silver nanoparticle, which are three-dimensional in the PBA matrix, which serve as support. There are different methods of synthesis of nanoparticles, top-down approach and bottom-up approach. What I do in the top-down approach, the standard is solid state, cutting of one mineral gas. Example is photolithography.
Hello, hello. Can I interrupt?
Hello.
Inder? I think
Yes, yes, ma'am.
you're not on the presentation mode. We can just see your first slide. Can you put the slides on the presentation and cover the slides as you speak?
Yes, ma'am. Just a minute.
Yeah, thank you. Please go on to presentation mode.
Yes, ma'am. Yes. Just a minute. Ma'am, now it is possible, ma'am.
Go on slideshow. Are you on slideshow? Slideshow from current slide or
yes, yes.
You want to whichever slide? Yeah, current slide is also okay. Yeah, go ahead. Your slides. I can see your slides. I think you're sharing, but cannot see your slides. Others can see.
No.
I think there is darkness there. Go on to some. You are presenting, but I cannot. Your point presenting.
Hello, Ravinder.
Yes, sir. Slide 2 page, sir.
Yeah, yeah. It's come up. So you just have to go to slideshow and do from current slide or whatever.
Yes, ma'am. Yes, ma'am. Close. Wait a minute. Sir, wait a minute.
Hello.
Yes, sir.
You just put
Yes, sir.
on your laptop.
Sir, I'm doing that, but entire screen is not possible.
Okay.
So you
Presentation mode also you can present the way it
present
was.
the window. You only present your window. Don't worry.
Yeah, let it come the way it's coming. Let it be like this. Just go on to the next slide when it is time to go on. Okay? You can see it. Oh, yeah.
Now it is okay, ma'am?
Yeah. Yes.
So types of nanomaterial. According to the dimension, we have the different type of nanomaterial. We have the 0 dimension, 1 dimension, 2 dimension, and the 3 dimension. In the 0 dimension nanomaterial, the dimensions are measured within the nanoscale, where no dimension are larger than nanomaterial. Such type of nanomaterial are said to be a 0 dimension nanomaterial. For example, is nanoparticle. In 1 dimension nanomaterial, 1 dimension is outside the nanoscale. That is, example of such type of nano dimension is nanotube and nanorods. In 2 dimension nanomaterial, 2 dimensions are outside of the nanoscale. Example of this 2 dimension nanomaterial is graphene, nanofilm, and nanolayer. In the 3 dimension nanomaterial, the 3 material are not constrained to the nanoscale in any dimension. This class, why this will not happen? Because it contains a bulk powder, bundles of nanowires, and the nanotube. In the present, what I heard, I grow the silver nanoparticles which are 3-dimensional in the PVA matrix. They serve as support. Next, there are different methods of synthesis of nanomaterial that are top-down approach and bottom-up approach. Now, what is top-down approach? In the top-down approach, the starting material is solid state. It refers the slicing or the successive cutting of bulk material to get a nano-sized particle. Now, in the physical method, come under the top-down approach. For example, is photolithography. In the bottom-up approach, the starting material either gaseous or liquid state. It refers to the build-up material from bottom, atom by atom, molecule by molecule, of a nucleation, and that result in the formation of nanoparticles. So, physical method and biological method is all method come under the approach. Physical vapor deposition, example of chemical technique, that is electrolytic deposition, mostly are using in the Solvay method. Biological method are totally based on the bottom-up approach. The difference between the chemical and the biological method is used for reducing and keeping stabilizing agent. In the chemical method, what we are using the strong chemical reducing agents such as sodium citrate, ascorbic acid. These are utilized, whereas PBA, polyvinyl alcohol, PBP, PAG, these are some chemicals, sodium borohydrate, etc., used as a stabilizer. The reducing and stabilizing agent for the biological method are provided by the reducing enzyme or secondary metabolite, which are present in the leaf extract or the different part of the biological plant. In the study, what I have taken the aloe leaf extract, which are utilized of nanoparticle. Now, method of synthesis of nanoparticle. We have the 3 methods for the synthesis of nanoparticle, that is physical method, chemical method, and the biological method. Now, what are the difference in physical method, chemical method, and green method, that is known as the biological method? In the physical method, they are the time and energy consuming. They also require the high temperature and pressure for synthesis of nanoparticle, whereas in chemical method, they are simple, inexpensive, and low temperature. But uses of toxic as reducing and stabilizing agent make it harmful. In the case of green method, it is very easy, efficient, and eco-friendly. Eliminate the use of toxic chemical, consume less energy, and produce a safer product. So, why silver nanoparticle? As we know, silver nanoparticle is well known for its antimicrobial property, and it resists the growth of microbial and protect the region of any contamination. In spite, the advantage of silver nanoparticle is in a certain amount it is toxic if the particles are prepared using chemical reducing agents such as hydrazine, sodium borohydrate, ascorbic acid, trisodium citrate, DMF, and ethylene glycol. It also adds the toxicity. So green chemistry can be used for the synthesis of nanoparticle. Now, what I done to grow in situ silver nanoparticle in polyvinyl alcohol, that is thin film matrix, using aloe vera leaf extract. Now, what is PVA? PVA is synthetic biodegradable material used in coating Using coating in plant structures, one dressing, and food packaging material. The physical property make it biocompatible with minimum cell addition. This draw us to develop a thin film of PVA matrix embedded to the silver nanoparticle with the greener method as to explore the applicability in the field of food packaging, wound dressing, coating in the healthcare industry. Hence, in this study, what I focus in situ growth of silver nanoparticle in PVA thin film matrix using aloe vera leaf extract as a bioreducing agent and characterization of structure. Further, we emphasize the biocompatibility study of the L929 and HA-CAT cell line of the material being produced and its potential as an antimicrobial property. Now, what are the methods for the preparations of PVA silver particle thin film? Now, just we have to take a 4 gram of PVA granule that is dissolved in 100 ml of distilled water at 80 degrees Celsius for 20 minutes on hot plate ventilator. The cell was cooled down at low temperature, room temperature. It is the 1 gram fresh aloe vera and mix manually with the glass rod. Next is and 1 ml of 100 millimolar silver nitrate stock solution was mixed and the beaker was exposed to the sunlight to synthesize the PVA incorporated silver nanoparticle. For the fabrication of thin film of sample solution, we have to use a clean, grease-free glass lamp that size is 15 into 20 centimeter. The sample is then poured into the glass lamp and were allowed to spread uniformly. The glass lamp were then cooled in the oven adjusted 35 degrees Celsius for 2 hours to get a film of PVA silver nanoparticle. Now, what is the characterization? How we can identify that silver nanoparticle is synthesized or not? So we have the different parameters to know the synthesis of silver nanoparticle in the PVA matrix. The first one is diffuse reflectance spectroscope. The reflectance spectra analysis was performed by the UV spectrophotometer. Just we have to change the reflecting measurement between the range of 190 to 1100 nanometer for the reflectance. UV visible analysis of PVA, plant track, PVA plant track, at the level of 300 to 800 nanometer. Now, nanoparticle tracking analyzer, that is NTA, that is basically based on the light scattering technique, allows the nanoparticle to be suspension on particle by particle basis. ICP and AES are basically used to know the estimate content of silver or the matrix. Second, the first plate double-sided carbon tape struck over 1 millimeter of the glass evolution casted to get image in the black scatter mode. XRD are basically turned to know the structure of the nanoparticle, maybe it crystals or amorphous. An antimicrobial study to test the efficacy of the microbial agent of the material being produced, it was accessed by the agar diffusion method. Microorganism they are chosen is S. uranus 25923 E. coli, E. pneumoniae, diploid fungus, has been swept uniformly. The Petri plate were incubated for 37 degrees Celsius for 24 hours and checked for the microbial growth. Biocompatibility testing, first one that is cell culturing. The normal mouse fibroblast, that is L929, and HA cell line were obtained from the National Center of Cell Science, Pune, and used for the biocompatibility assessment. Now, we also done the biocompatibility test for the PVA film. It was found the cytotoxicity of the film hospitalized or L929 cell by assay using the rubidium iodide for quantitative analysis. After attaining 70% confidence, harvested using 0.25% trypsin in 1 millimolar EDTA solution. And further, the pellet at 1000 RPM for 5 minutes, then resuspended in complete media. Another parameter is cell imaging. Further, the cell was imaged using propylium iodide, that is sigma average. The stained dead cell, thereby confirming the biocompatibility through the quality analysis. Now, result and discussion. By using the UV visible analysis, if the single peak are observed at 420 nanometer, that indicate the synthesis of nanoparticle. The appearance, absorption, and wavelength indicate the formation of spherical SMP, which is according with the TEM observation. If we get a single peak, that indicate the silver nanoparticles are in the spherical form, which can be seen over here. A single band is obtained, and this band is something related to the spherical size of the nanoparticles. Relatively narrow absorption peak of the sample are indicated for the relatively narrow size distribution and good dispersion of silver nanoparticle in the polymetrics. In present analysis, single band is observed at 5, indicating a spherical nanoparticles. Now, what is the result of TEM analysis? TEM analysis is mostly referred to the diameter of the particle as well as cell around it. TEM clearly demonstrates the formation of spherical silver nanoparticle homogeneously distributed across the polymetrics at a lower magnification where the density of silver nanoparticle are seen. TEM images, you can see the figure 3A and 3B show the presence of fairly spherical nanoparticle with various morphology. Silver is observed at the range of 20 to 50 nanometer. A bundle of sample nanoparticle with clear gain boundary can be seen at the range of 50 to 100 nanometer. The TEM at a higher resolution also reveals that each particle or cluster are not in the physical contact and separated with uniform interparticle distance in the polymetrics. This can be further confirmed by using the sharp Bragg reflection in XRD spectra. This is the XRD, X-ray diffraction analysis. The scanning range was 2 theta was selected from 10 to 80 angle. Here the XRD pattern clearly showed that silver nanoparticles are formed by the reduction of Ag iron by the leaf extract are crystalline in nature. XRD showed distinct diffraction peak around 38. 38 degree was the confirmation about the silver nanoparticle. This we have to compare with the standard data of the index number 1. Broad peak signifies smaller particle size and reflect the effect due to the experimental condition on the nucleation of growth of crystal nuclei. Now, this is the result of SAM analysis. SAM provides further insight into the morphology, size detail of the silver particle. The finding of the same analysis revealed that SMP were found to be embedded into the PBA film. Image figure show PBA film got aggregated to biester when bombardment with beams of electron, which can be easily seen in the figure. The result showed that the silver particle is aggregated to silver particles were well dispersed evenly over the polymetrics. ICP analysis. Silver incorporation in the PBA film was using analysis. It was found that 4.33 microgram of silver was present in 1 mg of the film. The NTA analysis gives an accurate estimate of the size distribution, and the concentration of the bio-reduced nanoparticle. In present case, the sample was in 1:4. The result is 49 nanometer with the standard deviation of 31 nanometer. The concentration photosynthesis nanoparticle was found to be 3.70 into 10⁸ 8 particles per ml. The observation size can be true as it is stated to be conformation with the reflectance spectra measurement. This is cell compatibility and morphological analysis using the PVA dye after treating with PVA film. It was shown L929 and HA8 cell line. This figure demonstrates the PVA film proved to be a biocompatible because there is no cell death or any change in the cell morphology. Various researchers have already proved that PVA are biocompatible and hemocompatible, hence different type of biomaterial and the surface coating are made up of PVA. This is antimicrobial testing. Zone of inhibition shown by antibiotic ticks. There is no growth under the PVA SNP film by E. coli or S. uranus. This is zone of inhibition by the antibiotic ticks by using the PVA SNP film. CMOs does have no deleterious effect. PVA itself is highly biocompatible. Thus, when combined with strong bacterial agent, the application potential is likely to enhance investigation of bacterial efficacy against the four pathogenic organisms, confirm their sensitiveness of the bacteria toward the material or film by film being produced. And ability of the material to fabricate on coating onto different surface is likely to enhance the application. The feasibility is handling and preparing the film make it potential object for the use in wound dressing, food packaging, cosmetic industry, and for the various application in nanomaterial and this is some and thus end my presentation. Thank you everyone. Any question regarding this topic? You can ask me. Hello.
Hello, Mr. Farooq.
Yes, ma'am.
It's a nice presentation.
Yes, ma'am.
The only thing is because of lack of proper internet connection, there was a sound which we could not hear properly, but your work was very good is what I could see. My only question to you was, is there any reason why you selected aloe vera, which is so much work to plant on?
Ma'am, I'm selecting the aloe vera gel because it is easily available and as compared to other plant extract, it is very simple to take the extract of aloe gel while preparing the nanoparticles, silver nanoparticles. So extraction is very compared to other and it also have a very good medicinal property.
Yeah. Did you simultaneously also try any other plants, and did you find difficulty if you have tried? Or
Yes, ma'am. We are definitely trying various types of plant for synthesis of silver nanoparticle as well as other nanoparticle.
Okay, but so did you find any difficulty and variation when you were working with the aloe vera and you were working with other plants?
Yes, ma'am. Because in the other plant, we have to either silicate or we have to boil to make the solution. But in the case of aloe vera gel, slightly we have to take a leaf and take the extract of the gel. So as compared to other plant material, this was good for taking the extract.
Okay.
It is easy to take the extract and synthesis of nanoparticle as compared to other plant material.
Correct. And how much time do you think you required for all this work which you have presented today?
It is nearly around 3 to 4 months for doing this.
So in 3 to 4 months, you have completed your entire nanoparticle making and doing all these assays and estimations?
Ma'am, that testing only required me more time, around 2 to 3 months. Characterization and testing it required 2 to 3 months. This overall 4 to 5 months is applicable for doing this all research. around 4 to 5 months.
Okay. Okay, good, good. Thank you, thank you. Any
Thank you.
other will come up.
Thank you, Parminder.
Thank you, sir.
So now we shall move on to our next presentation. Ravinder, thank you so much. And our next participant, our next presenter is Mr. Pramod Kuke.
Yeah.
Mr. Pramod Kuke.
Yes, ma'am.
Yes, you can begin with your presentation.
Yes, ma'am. I'm sharing my slide.
Yes.
My screen is audible, ma'am.
No, your screen is not visible.
Sorry. Pramod, what is happening? Start your presentation. Hello.
Mr. Pramod, are you there?
Yes, ma'am.
We are running short of time, so I request you to be a little quick.
That's not perfect. Now the slide is visible.
Now your presentation is visible. Start.
Yes, yes.
Myself, Mr. Pramod Kuke. I'm doing PhD under the guidance of Professor Dr. Ahuja ma'am, and my paper title is In Vitro Multiplication for In Vitro Phallus Induction in Medicinal Plant Saludora persica. Introduction. Saludora persica belongs to the family Saludoraceae. Is an important shrub, 4 to 6 meter tall, in short trunk, wide bark and small green leaves. Saludora persica is the Ayurvedic system of the medicines. Is reported in have a pointal activity for a dental gravenus. It is called as the miswak tree and root or big.
Pramod, your slide is not moving.
What, sir?
Slides are not moving.
ये slide आपका visible नहीं हो रहा है next वाला.
स्लाइड शो पे जाके
slide show करो, उसको next करो.
Present from current slide.
Slide is visible, ma'am.
Slide is visible. Just put on slideshow so that you put on the next slide now. Only the first slide is visible. Can you go on to the next slide? Yeah.
Saludora persica belongs to the family Saludoraceae. It is an evergreen shrub. They have nothing but the 4 to 6 meter in tall and the short trunk, wide bark and small green leaves. Salvador persica is the Ayurvedic system of the medicines. They have reported have a point activity of the dental evidence. It is called as the miswak tree. And the miswak toothpaste is also utilized on the dental cleanings. The root and twigs of this tree have been used for the teeth cleaning since ancient times. It is one of the most commonly used in the medicinal plant and oral hygiene among the global Muslim community. Miswak has an analgesic, anti-region, and anti-inflammatory property, making is an effective treatment of the primary periodontal diseases. Evidences suggested the miswak being activities against in the thermal stimuli compared to the chemical ones, focusing on the physiology of the response of the terminal stimuli are via skin pains receptors, whereas it is the chemical stimuli as in the response via inner visual receptors. Sorry. Thus, it is found in the miswak respond to the peripheral pains of the not in the vesicles. It is the applied to the oral mucosa. It is set of the roughly on the oral pains also. Experiments on the means of the laboratory provide the miswak has the moderate analgesic effect of this related to the interaction to the peripheral optic system of the addition and being noted on the potent practicing miswak regularly had on the low incident of their toothache compared to their toothbrush use. Medicinal and chemical properties of the miswak. Miswak chewing stick is an dental care tool achieved on the mechanically by the ability of its fibers and reach into the and also chemically the reaches of its phytoconstituent, which is the unique in their complexity and biological activities of Salvadora persica. Demonstrated of the reaches of the Salvadora persica from the minerals and phytochemical component related to a dental care. Many studies focusing on the extracted chemicals such as the beta-cholesterol, glycosides, and organic compounds like pyrrolidine, pyrrole, and piperidine derivatives, which is biologically active and is described as remedies for the joint pain, splint tumors, granules, skin diseases, and stomach ulcers. Those are the very common diseases. Fluorides on flavonoids miswak extract was also shown in the important elements the realizes teeth and among which show in the antimicrobial activities of mouth pain also, or mouth care also. Miswak soaps is reported in the stimulant of salvia production of the pH buffering active capacity, ability to remove the dental plaque bears, remolarization of the enamel layer of shown in the strong antimicrobial activities. Materials and methods used for present research work. Preparation of explant and sterilization. The explant like leaf, stem, nodes collected from the young health plant of Salvadora persica from different localities of Aurangabad region. All this explant was washed with running tap water for 5 minutes, followed by 70% ethanol for 1 minute, and finally with distilled water for 3 minutes. Surface sterilization of explant was carried out by washing with sterile distilled water for 3 minutes, followed by various concentration of mercury chlorides. Late explant is sterilized with 0.1%, whereas tail nodes with 0.2% of HgCl2. It was followed by 2 subsequent rinses with sterilization, double sterile distilled water inside of laminar airflow. All these experiments were cut into explants were cut into a small piece and inoculated on MS medium. Culture condition of all explants of investigated were treated with MS media, Morasaki and Scoop 1960, supplemented with various concentration of growth regulators. MS medium was fortified with the 3% sucrose and 2.5 to 3% of the clerical solidification of pH was adjusted to 5.6 to 5.8. The media were steam sterilized with autoclave in 15 lb pressure. Culture conditions. After the inoculations, culture bottles were transferred into culture rooms in 16 hour photo period, supplemented by white fluorescent cooled test cold tubes light temperature at 25 degrees Celsius plus minus 2 degree. At least 5 replicates rises for each treatment, and their data were recorded in the tables. Result and discussion of present research work. Total surface sterilization was analyzed by trial and error method. Surface sterilization of leaf and stem node explant were tried with 0.1 to 0.3% of HgCl2 for 3 to 5 minutes duration. The maximum microspheric culture and high regeneration percentage were recorded at 0.1% for leaf and 0.2% of HgCl2 for nodal segment. Aiding the gradient study, hormones MS media was found ineffective regeneration of shoot by the explant, shoot tip and nodal segments. Callus regeneration was achieved from a leaf explant from 240 to 1 mg per liter. Tribal greenish and whitish callus are formed. Nodal segment at the concentration of 2.0 mg per liter of the 240 yield tribal greenish callus are formed, whereas shoot tip at the concentration of 2.5 mg per liter of the 240 gives tribal greenish and massive callus. This observation table shows the effect of different concentration of 240 DAP kinetic on callus production. First of all, the leaf explant 240 is 1 mg per liter, formation of tribal greenish and whitish callus. Second one is the nodal segment for concentration of the 240 2 mg per liter. Tribal greenish or white callus are formed. Next, third one is the shoot tip. In case of the concentration of the 240 is 2.5, the tribal greenish callus are formed. This photoplate shows the result of different concentration of the callus induction. First one is the 241 mg per liter. Greenish callus are formed. Second one is the 242 mg per liter, tribal callus are formed. And third one is the 240 2.5 mg per liter of the greenish callus are formed. Of present research work, medicinal plants are collected from the treatment of many disorders. These plants are exploited at large scale. This plant need to use. If these plants are propagated through the modern technique like plant tissue culture, within a short time period, we can grow a large number of plantlets. Present work is useful for the development of the micropositional studies of Salvadora persica. And this technique is also utilizing for the conservation of medicinal and endangered plants. Next one, thank you. If any questions, so please ask.
Thank you, Mr. Pramod. Okay, it was really nice presentation and well presented also. You have made the slides and the pictures also very well. But
thank you, ma'am.
I am just thinking what is the question that I can ask you in this particular topic is you have already said that meswak, the plant, the Salvadora persica that you have identified, and you have already got the regenerated callus, and you've seen in different 2D the hormones, whatever you have used. Have you also used any other toothpaste which is of the similar kind, which is Ayurvedic toothpaste, like you have many other Vico turmeric and everything? Why only? Is it because a single plant is used there, or is there anything else that has come to your mind?
Ma'am, I have selected Salvadora persica. Why? Because of they have this such type of the antimicrobial or anti-cancerous activity of mouth cancer.
Okay.
That's why I am selected this plant for my research work.
Okay, and you have already got a good regeneration of callus also, although.
Yes, ma'am.
Yeah, that's very good. If you work more on it, maybe you'll get the entire whole plant character.
Yes, it's my next step to regenerate whole plant and hardening and conserve it.
Conserve it, and also you can even try at cellular level when you are getting the callus from the cells of the callus. Can you the active constituents generated by just a medium in a fermenter? Then you don't even have to grow the plants. You can grow the cells and you will have the active constituents growing there, and you can pull out the active constituents and make a toothpaste rather than using the plants, can't you?
Yes, ma'am. These are my next step of the research for phytochemical analysis of the callus, stem, root, all these things in vitro cultures also.
Yeah, okay, good, good. Nice work. Thank you.
If any question, anyone else? Anyone others?
Any others with any queries? Any questions? So since there are no more questions, thank you,
Okay.
Mr. Pramod, for your
Thank you.
presentation.
Thank you, ma'am.
It was a good presentation.
Thank
I would now like to call upon our next presenter, Dr. Preeti Patil. Dr. Preeti.
Hello, am I audible?
Yes, Dr. Preeti, you are audible.
Pramod, could you just stop your presentation so that I can present my...
Mr. Pramod, okay.
You can start.
Can you stop your presentation?
Oh, yes, ma'am.
As well as put off your microphone.
Yes, and stop my presentation, ma'am.
Thank you. Dr. Preeti.
Yes. Is my screen visible?
Yes, your screen is visible.
Okay, so welcome all of you. National Conference on Different Advances and Opportunities in Botany. So the today's topic of my presentation, paper presentation is Biodiversity Studies of Myxomycetes in Alibag Taluka, Raigad, Maharashtra. So the present paper deals with the biodiversity assessment of Myxomycetes from Alibag and its nearby region. In this paper, I have listed 27 Myxomycetes species which are collected from various regions. And during this survey, I found that the Kankeshwar forest is found to be the best reservoir for this species. Introduction. Now, what are Myxomycetes? Myxomycetes are the eukaryotic organisms which are mostly overlooked in nature. Previously, they were studied by mycologists, and now current classification placed them in superclass Amoebozoa. So they follow the uh, so they follow the International Code of Zoological Nomenclature. Now, currently hundreds, 1,000 species were known, and they are cosmopolitan in nature, and they are mainly found associated with decaying wood, leaves, and aerial litter. So wherever there will be moisture, they grow. Now, why Myxomycetes are overlooked in nature? So these two pictures says everything. You can compare the size of the Myxomycetes. Here you can see, this is a Google image, this is an image taken by me. So you can see how small they are. They are below 1 centimeter, or you can say 0.1 mm to 0.2 mm. Few species may measure up to 1 centimeter also. And why they are overlooked? Because the place where they grow in nature. So these are the places you can find these species in decaying woods, the litter, but they always you can always find them underside of the wood. Now, what are the components of Myxomycetes? This is the fruiting body of Myxomycetes. You can see it has a stalk, it has a hypothallus. This fruiting body structure, it has a covering which is called as peridium. Inside peridium, a mass of capillicium is there, which helps in spore dispersal. Inside spores are present. So these are the different components of Myxomycetes. Now, the life cycle of Myxomycetes. This is a fruiting body. The spores are liberated. They undergo germination. During germination, they produce two types of cells. Some cells are amoeboid in nature. They are called as myxoamoeba. And some cells show flagella, so they are called as swarmers. Myxoamoeba fuse with each other, swarmers fuse with each other to form zygote, and it undergo mitosis to form plasmodium. These plasmodium move from one place to the another place in search of bacteria, and under favorable conditions, they sporulate to form fruiting body. So previously they were also called as plant animals because the character of producing spore is similar to plants, and the locomotion which is shown by the plasmodium is similar to animals. This life cycle which I have shown you, I have in my PhD thesis work, I have my main aim was to culture them in agar. So this life cycle I have completed in agar culture also. For this species, Dicea subspecies, this is the fruiting body you can see on the leaf, and these are the different stages on the agar. Here you can see the myxoamoebas and swarmers. They are fusing with each other to form plasmodium. These are young plasmodium. Here you can see this is the mature plasmodium, white color in nature, and these plasmodium strands under microscope looks like this. And here you can see the fruiting bodies, they are formed outside the Petri plate. So this study was the first spore to spore agar culture of Dicea subspecies. It was the only second species which was cultured till now. So from India and from world, this was apparently the first study. Here you can see the different stages of sporulation. I have collected the plasmodia from Chondi. This is a place near to Alibag. Here you can see how the fruiting body are formed. This all procedure takes place in a single night. Here you can see the early stage of sporulation. After few hours, you can see the growth of the stalks here, and then you can see this beautiful color of fruiting body. And here you can see the color change in the fruiting body. This is Physarum globuliferum. So in such way, the whole procedure of sporulation occurs. Now classification of Myxomycetes can be divided, or it is divided into two classes, exospory and endospory. In endospory, different 5 subclasses are there. Now this is the Concassia forest, which I told you that I found it best reservoir for the Myxomycetes. This is the front side. You can see the trail. You have to climb 1700 steps to reach to the top. And this is the back side of the Concassia. Now how the collection is made, actually this is the fruiting body of Stymnoites. It is on the decaying log. So you have to cut the substratum, along with the fruiting body, and then you have to stick them on the paper, and you can then put this fruiting body along with the substratum in the plastic boxes or in the card boxes. These are two. Here you can see it is on the leaf, and here you can see it is on the wood. So this is my personal collection. Here you can see how the boxes are prepared. You can see here paper on this such paper, so that you can easily lift that species with the help of paper. And after preservation, you can maintain the important data on the books, on the box. For example, the date of collection, the place where you collected the specimens, and if you know the name of the genus and species, you can put them. So this is my personal collection. Identification was made on the basis of morphological character, and for this, the literature is used. Now, results and discussions. So these are the different species that I have collected from different locations. All these, most of the regions from this is the coastal town. Here you can see the different species like Hemiphyta circula. I will directly show you the pictures because they are so beautiful. So these are the different list of different species of Myxomycetes. I will go directly into the images. So this is Arteria denudata. You can see on the wood. Again, this is Arteria ovulata. Again, this was found on the wood. This is Arteria cinerea. This is again Arteria denudata. This is Serratiomyxa fruticulosa, which belongs to class Endospory. This is Criberia. This is again Criberia infected with different fungi. You can see other Ascomyces also here. Some Pezigels member also. This is again Criberia. This is Diderma hemisphericum, which I collected from my college campus only. This is Dicea subspecies that I have shown you earlier. This is Diderma species. These are some Didymium species. Here you can see the stalk and the peridium. This is the stock view of same species. This is the Fuligoseptica. As I told you, these are Plasmodium. Here you can see how they move from one place to another, and after favorable condition, they form the fruiting body. This is the fruiting body of the same Plasmodium. This is the Plasmodium of Fuligoseptica. This is also called as dog's vomit, and this is the fruiting body of the same. This is the zoom photo of Fuligoseptica. Here you can see the spore mass. This is Hemitrichia circula, same Hemitrichia circula, but this is a somewhat zoom picture. This is again two types of fruiting body can be seen on a single leaf. This is a somewhat athelium fruiting body without having stalk, and these are having stalk. This is Lamproderma. Here you can see how small they are. Actually, these Myxomycetes need a very good lens, for example, 100 mm lens, so that we can capture these Myxomycetes. This is Physarum globuliferum, as I told you earlier. This is Physarum lilium, yellow color, orange color fruiting body and white color stalk. Again, it is present on a leaf. This is again Physarum species. This is Physarum viridi, yellow color fruiting body. Here you can see this. This is Physarum oblonga. You can see this shape of fruiting body makes the name oblonga. Here you can see this was found on a coconut leaf base. This is Stymnoitis exifera. This is about 1 centimeter. Here you can see the stalk. This is Stymnoitis fusca. This is Stymnoitis flavogenita. This is Stymnoitis smithae. This has a good story. I have found this on my colony wall. Actually, they are generally prefer the places where the moisture is there, but I don't know how they occur here. I don't know, because it is a cement wall, and I have took the below photo. So these are looking like a hanging Stymnoitis species on the wall. This is a somewhat ghostly photo. Stymnoitopsis hyperopta. This is again related to Stymnoitis only. You can collect this fruiting body. You can collect Myxomycetes in the form of Plasmodium also. Here you can see the Plasmodium. This I have collected from the forest, and I have transferred it to the tissue papers, filter papers, and on agar. This is again the Plasmodium in the forest. This is the Plasmodium on agar plate. Here you can see this is the Plasmodium on wood. This is the Plasmodium on water. They move. This is called as advancing fan. Whenever they get bacteria to eat, they will move in that direction. Again, this is a Plasmodium in the nature. Here you can see the Plasmodium. Again, this is Plasmodium. These are different Plasmodium. This is okay. Here you can see the early sporulation. This will get converted into this. Here you can see how the white color structure gets converted into black. This is actually Dicea subspecialis that I have shown you earlier. This is again Physarum globuliferum. I have brought this wood with Plasmodium, and next day I found this fruiting body. Sorry, just a minute. Okay, so what are the importance of slime molds? Slime molds are they have anti-cancerous, they have antifungal, they have antibacterial properties. The one of the compound was extracted in 2015 named polysapin from Physarum polysapinum, which was used to treat breast and brain tumor. About 100 secondary metabolites have been extracted from different slime molds. There people in Mexico used this fruiting body. They fry it and they eat, and they are also important secondary separators. Where we stand in Myxomycetes research, in India, the research is only restricted to taxonomy, and the Indian Myxomycetes research can be divided into 4 phases: period 1, period 2, period 3, and period 4. And from Maharashtra, only these persons have done taxonomic studies in Myxomycetes. Dr. R.N. Mishra is my guide. He's retired now, and they have covered Mahabaleshwar region, Pune region, but most of the Maharashtra is still not covered. So conclusion, so among 1,000 species, only Physarum and Didymium were the only model organisms, and so nothing is more known about this Myxomycetes species. So it's our aim to collect these species, to preserve them, and to study them so that we can understand the biology of these organisms, and we can explore these organisms so that we can utilize it to serve the human race in future. So this is my future plan, where I will be dealing with biochemical analysis of Myxomycetes. These are the bibliographies. And what are the lessons to take home? That these are the beautiful organisms. You can find it in your backyard also after rainy season. And these are the best models for the photographer. Thank you.
Excellent. Dr. Preeti Fatty, it was very beautiful to hear about Myxomycetes, where not many people are working on. And you are doing an excellent work, and further biochemical analysis is really the need of the hour, because you will find many, many more metabolites, secondary metabolites in all these species of Myxomycetes that you have found.
Yes, ma'am.
Carry on more research in the biochemical assay, because you've already found so many of Myxomycetes, while others can follow your steps and do a lot of research.
Sure.
Excellent work. Good.
Thank you, ma'am. Thank you so much. Any questions?
Thank you, Preeti ma'am.
Yes, sir.
Nice presentation.
Thank you, sir.
Thank you, Dr. Preeti.
Okay. Thank you, ma'am.
As there are no queries, we will move on to our next presenter.
Yes, of course.
Thank you, ma'am. So I call upon our next presenter, Dr. Hasmukh Bhai Karadi. Dr. Hasmukh Bhai Karadi.
Maybe Hasmukh Bhai not presenting.
What, sir?
He is not presenting.
Oh, okay, sir. So no problem. So we can move on to our next presenter, Ms. Shilpa Chavan. Ms. Shilpa Chavan.
Hello. Hello.
Yes, we can hear you.
Just a minute.
Hello, Preeti ma'am.
Yes, sir.
Start your presentation, please.
Just a minute. Sorry, sir. Really sorry.
उनको हंसी आ रही है प्लीज
Yes, sir. Stop your screen.
So it is my screen, Shilpa Chauhan.
Okay.
I'm sharing my screen.
No problem.
Hello, myself Shilpa Chauhan. I am doing PhD under the guidance of Professor Dr. N.B. Pandure, Department of Botany, Dr. Bamu, Aurangabad. My topic is in vitro callus induction of African baobab tree, Adansonia digitata.
Hello.
Hello.
Your screen is not visible.
नहीं है 1 minute sir.
Presentation is not visible.
I think we are facing some technical errors. We will wait for a few minutes.
Yes, wait for a few minutes. That's one thing. Ask me something. Promote offender. Promote offender. त्याला सांग हे प्रेझेंटेशन पुढचं प्रेझेंटेशन करून त्याच्यानंतर शूट करावं. हां मॅम काय होते करतायत ना आज त्यांना पोस्ट त्याच्यामुळे आली.
Sir, screen is visible.
No, the screen is not visible.
Your screen is not visible. Promote.
Sir, presentation now, na?
Yes, yes.
Yes.
Enter screen, na?
Yes. Present now and share your screen, please.
Enter your screen, but sir.
Is it visible, sir?
Not yet.
No, let's see.
You can mail it to someone and
Just a minute, ma'am. Is it my screen visible? Hello?
Yes.
Yes,
Hello?
yes, yes.
Yes,
Is it
yes,
visible?
yes.
Hello, myself Shilpa Guru Chavan. I am doing PhD under the guidance of Dr. Narayan Pandure sir, Department of Botany, Dr. Baba Saheb Ambedkar, Marathwada University. My topic name is in vitro careless induction of African baobab tree Adansonia digitata. Introduction. Adansonia digitata, it is also called as baobab, is a fascinating tree of African savanna and known for its enormous size and distinctive appearance. Baobab is one of the 8 species of the genus Adansonia, but it has until recently been considered the only native species on African mainland. The tree may reach up to 25 meter in height and may live for hundreds of years. It is very important in the economy of rural and urban people for food, shelter, medicines, and numerous other products. It is also used in cosmetic products. It is also resistant to fire and survive well in dry climate. The leaf, bark, and fruit are employed as foodstuff and medicinal purpose. Uh, it has numerously health benefits which can be related to the presence of bioactive compounds like terpenoids, saponins, tannins, etc. that are isolated from its various parts like leaves and fruit. The tree has mythological significance and is also known as Kalparvriksha in India and elsewhere. Various plants parts such as leaves, bark, and fruit pulp have been traditionally used in immunostimulant and anti-inflammatory, analgesic, and in the treatment of fever, diarrhea, gout, dysentery, hemoptysis, tuberculosis, microbial infection, and worms. Medicinal and chemical properties. Adansonia digitata is one of the largest and long-lived tree in the world. It is evident that the baobab is nutritional medicinal resource. It is tree that can provide food, water, shelter, and relief from sickness. Every part of the plant reported to be useful. The global demand for the baobab has increased dramatically in as more sectors as cosmetic industries and used in pharmaceutical industries due to its fatty acid content known to have beneficial effect when applied into skin. Baobab fruit pulp have been approved by a statutory body for use in certain nutritional products. It have various usage used as astringent, demulcent, diaphoretic, diarrhea, dysentery, and hemoptysis, pneumatic pain, inflammatory ulcers, intermittent fevers, antitrypsosome, antibiotic, anti-cancer, diuretic activity. The tree in thus named as the small pharmacy or chemistry. Materials and methods, preparation of explant and sterilization. The explant like leaf stem node was collected from young healthy plant of Adansonia digitata from different localities of Aurangabad region. All these explants were washed with the running tap water for 5 minutes, followed by 70% ethanol for 1 minute, and finally with distilled water for 3 minutes. Surface sterilization of explant was carried out by washing with sterile distilled water for 3 minutes, followed by various concentration of mercury chloride, HgCl2. A leaf explant is sterilized with 0.1%, whereas stem node with 0.2% of HgCl2. It was followed by 2 subsequent rinsed with the sterilized double distilled water in laminar airflow. All these explants were cut into small pieces and inoculated on MS media. Culture media. All experiment of investigation were tried on MS media, supplemented with various concentration of growth regulators. Medium was fortified with 3% sucrose, 2.5 to 3% of clarigar for solidification, and pH was adjusted to 5.6 to 5.8. The media was steam sterilized in autoclave under 15 psi and 121 degrees Celsius. Culture condition. After the inoculation, culture bottle were transferred to culture room under 16H. Photo periods supplied by cool white fluorescent cool tube light and temperature at 25 to 2 degrees Celsius. At least 5 replicates raised for each treatment, and data were recorded in table. Result and discussion. Standard protocol for surface sterilization of explant was analyzed by trial and error method. Surface sterilization of leaf and stem node explant were tried with 0.1 to 0.3% of HgCl2 for 3 to 5 minutes duration. The maximum microbes free culture and high regeneration percentage was recorded at 0.1 percent for leaf and 0.2% of HgCl2 for nodal segment during the present study. The hormone free MS medium was found effectively regeneration of shoot using both explant leaf shoot tip and nodal segment. Maximum amount of callus induction are achieved in the different concentration of media. Callus regeneration was achieved from leaf explant in MS medium from 0.5 mg per liter, TDS 7.0 mg per liter, AA 2.0 mg per liter, BAP fragile greenish and whitish callus are formed. Table effect of different concentration of BAP, NA, TDS on callus formation in MS media. TDS had 0.5, NA 7.0 and BAP 2.0. It is seen in a greenish color. Callus are found in a greenish color. It is found in these two concentration different media. Conclusion: medicinal plant of Adansonia are collected for treatment of many disorders. These plants are exploited in at large scale. These plants need to be conserved. If these plants are propagated through modern techniques like tissue culture, then within short time period we can grow large number of plantlets. A present piece of work is useful for developing rapid micropropagation of Adansonia plant. Thank you.
Thank you. Nice.
Hello.
Yes. Does anyone have any query? Nice presentation, Ms. Shilpa.
Yeah, nice presentation. Good one.
Thank you.